Agriculture, Indore. So it gives me an immense pleasure to announce and to tell you all that we are having a very dynamic personality, Dr. S.V. Sai Prasad sir, Principal Scientist, Indian Institute of Rice Research, Hyderabad, as a chief guest for today's function with us. So it is a start of any program. It is always incomplete without God's praise and spirituality. So I request to enlighten the lamp and Saraswati Vandana in order to start the function. I request Sri Shadab Sheikh sir to please play the online clip to lamp lightning and Saraswati Vandana.
सोमा ज्योतिर्गमय नित्यमृतमृतं गमय ॐ शान्ति शान्ति शान्तिः या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Thank you, sir. With the praise of God, let's start the function. So if we desire to create the environment where education system should be perfect or will be perfect, for that reason, we need to update our knowledge. We need to update the knowledge of faculty so that the capable atmosphere is created where all the faculty members can work together to accomplish their task both personally and professionally. For this reason, the faculty development program has been conducted every year by Sri Vaishnav Institute of Agriculture, Indore. The whole program is designed to provide the opportunity for all the faculty members working in various institutes in India as well as abroad to come and experience the environment, the curriculum, the mode of teaching and learning in our institute. This program will help to bring the required changes in the attitude, skill, and knowledge of all the staff members and faculties. This program will cover the various topics such as tissue culture techniques, hydroponics, mushroom cultivation, productive cultivation, and poultry farming by various eminent speakers. So, as we all know, in Indian tradition, we always says that Atiti Deva Bhava, means our guest is considered as our God, and welcoming them seems to direct praising to the God. So may I request Dr. K.N. Guruprasad sir, Dean, Faculty of Agriculture, Sri Vaishnav Institute of Agriculture, Indore, to please welcome our today's chief guest, Dr. S.V. Sai Prasad, by some welcome words. Sir, please.
Thank you, Archana. Chief guest, Dr. Sai Prasad, our guest speaker of this morning session. We have two speakers, Dr. Ashok Jadhav, I can see on the screen, and I guess Dr. Pooja Pandey is the other guest for today's morning session. So Dr. Sai Prasad is well known to us, and he is very familiar with Sri Vaishnav Vidyapit Vishwavidyalaya. But as a part of my duty, on behalf of the university and on behalf of Institute of Agriculture Science, I welcome him for this inaugural session. This faculty development program has Agri Entrepreneurship as its theme for and has invited experts who are working in this field of agri entrepreneurship. So the participants will be exposed to various progress and various types of agri entrepreneurship that is possible in India. Several times we hear about the international programs of sustainability. Even sustainability has taken the central stage now in every developmental program as far as industrial program. Now, in this context, agriculture science assumes an extremely important area for developing anything that is sustainable, since agriculture deals with sustainable development and sustainable innovations. Using agricultural materials for innovative ideas will automatically make it a sustainable approach without any extra effort, since all the agricultural products can be easily recycled and can be reused. Unlike other industries, where recycling, reusing, and also making it toxic free becomes a problem by itself. Any industry based on agricultural products will not have this problem, since they will be dealing with the natural products which are naturally degradable in any form. To that extent, agricultural entrepreneurship is going to play an important role in the future of every country, since United Nations has put the sustainable development as the platform for the further progress in order to uh, protect the climate, and also in order to produce less toxic substances through the industries. Most of the times we hear, particularly in India, that agro-based industries in the rural areas are not developing at a pace at which you would like them to develop. Now, this is for various reasons, because one of the main reasons may be the lack of infrastructure development in the rural areas to experiment with innovative projects based on agriculture. Now, this is an area that needs to be developed by the governments so that it gives a boost to the agricultural entrepreneurship. Agricultural entrepreneurship requires an all-round development. It's not enough only for the scientists to think of having innovative projects, but there has to be a supportive infrastructure development, especially in the rural areas, so that scientists and students of agricultural science can settle in the rural areas and still do agro-based industries. I hope this faculty development program will enlighten us about some of these ideas, which will be extremely useful for the participants in this program, and further transmittance of these ideas to their own students in their respective institutes. With these words, I welcome Dr. Sai Prasad once again, and over to you, Achan.
Okay. Thank you, sir.
Thank you,
Thank
sir.
you.
Thank you, sir. And personally, sir, welcome to this program. And I hope you will have a good time with us. So let's have a look on the work which is done by our chief guest, Dr. Yasvi Sai Prasad, sir. So Dr. Yasvi Sai Prasad is currently working as principal scientist, Department of Plant Breeding, at Indian Institute of Rice Research, Hyderabad. During his working tenure, he has attended more than 10 training programs. He is involved in developing 27 improved varieties of different crops such as durum wheat, braid wheat, fodder oats, field pea, lentil, and also is associated in development of 18 improved wheat varieties, including one durum wheat. He has played a lead role in developing 6 genetic stocks of durum wheat and associated in development of 14 braid wheat genetic stocks, which are registered at New Delhi. In team, he has developed 6 bio-fortified wheat varieties. Dr. Yasvi Sai Prasad has also identified diverse source of stem and leaf rust resistant durum wheat. Along with the research, he has also guided 2 PhD students and 14 MSc students. He has published more than 13 research papers and has been awarded with various awards by ICR and various research institutes at national and international level. In future, he is planning and breeding for high yielding, medium to late maturity rice varieties with tolerance to biotic stresses and good grain quality. With all the profile of Dr. Jasvi Sai Prasad sir, I think we all must be eager to hear from him, to know him, and to know more detail about him. So may I request Dr. Guru Sai Prasad sir to kindly throw some light on today's FDP program entitled Agri Entrepreneurship to enlighten all the participants of this program.
Thank you, Dr. Archanaji. Myself, Dr. Sai Prasad, presently working here at Indian Institute of Rice Research. And before that, I have worked for around 22 years at IRA Regional Station Indore. At that time, I have close association with Dr. K. Guru Prasad ji, who is like a teacher to me. And we are having a good relationships with Sri Vaishnavi Vidyapith Vishwavidyalaya, because that is not a separate institute, it is like a in-house. I used to feel that it is like a family to us. And I have learned a lot of things in that university as a member of, along with Dr. K. Guru Prasad ji. And that is a very good institute, Sri Vaishnavi Vidyapith. And I pay my respects to Honorable Vice Chancellor of the university, as well as Dr. Vinod Dhar and Dr. K. Guru Prasad ji, Achana ji, and my student and SRFs used to do Dr. Chavi Tiwari. I am very fortunate that you have given an opportunity to me to be as a part of this program. This Agriculture Development Entrepreneurship Development Program, which is a faculty development program which has been initiated by this university, is a very good opportunity for all the faculty members of different institutes who are working closely to agriculture. Nowadays, everything has become like a business. So a student who is joining in a BSc Agriculture, he must think like an entrepreneur, what he will do, what is his goal in future. And there are a lot of opportunities are there in agriculture. So I congratulate all the faculty members who have been involved in this training program at this institute, both personally, and they must professionally also be competitive, because nowadays it is a competitive world and everybody must take it as a competitiveness in every development program, because this is not a one-way development program from this institute, whereas the faculty members who came from different institutes, they must be a two-way interaction, and they must also come forward to tell that what are the lacuna at these universities, or how they can improve, and how their universities can be improved, their institutions can be improved, seeing the infrastructure developed by this university. Because agriculture department has been started in this university in a very recently, but within this few years, they have shown the development. Even faculty has been trained properly for teaching the students. Because Dr. Achana has told that they are giving some information about the tissue culture, poultry and protective cultivation. At the same time, they must also have some information about this genome editing, because nowadays the genome editing is also a new technique, biofortification and value addition. Unless you have value addition of your products, so you will not be competitive in your market. So every faculty member should always think out how to inoculate the entrepreneurship in the students who are studying the AGBSc, MSc and PhD students, how they can inculcate that entrepreneurship and how they can do the value addition of their products, agricultural products, and how they can able to establish the companies at the village level so that they can improve the infrastructure of the villages. I will give one example at Indian Institute of Millet Research in Hyderabad. They have started a small program of the Agri entrepreneurship for millets, because next year, 2023, is the millets year, International Millets Year. So for that program, they have started a small program some 4 or 5 years back by training few agri business graduates in entrepreneurship development. And they have supported financially with a small amount. And now I will give one example that Millets Dunia. There is one company called Millets Dunia. There is one entrepreneur called Dr. Pujita. She has got trained in this type of entrepreneurial development program at Indian Institute of Millets Research. She has been trained in the value addition of the millets and different forms of millets, how they can commercialize. And now she has started a startup entrepreneur startup called Millets Dunia. And now she has been marketing at both domestic as well as international level. So like that, I always suggest that whenever in this type of programs, faculty development programs, you must inculcate those techniques so that you can able to guide the students of agriculture, graduation, post-graduation, and these people, how they can go as an entrepreneur and how can they can show some jobs to the other graduates. As a team, they must work. In the same way, I will give another example. In East Godavari district of Andhra Pradesh, agriculture students from AG College, Agriculture College, Bapatla, some 4 or 5 friends, they have started and they have gone for training to CAE Bhopal, ICR Institute, Central Institute of Agricultural Engineering, Bhopal, and they are able to see what are the service equipments they can give to farmers on higher basis. And now they have started in East Godavari a company called as service providing to the farmers from sowing to harvesting, tracing everything up to marketing. And they are showing the ways how to market their products. So this type of total information, it must be a two-way interaction between the faculty from other institutions as well as the faculty from Sri Vaishnava Vidyapith Vishwavidyalaya. I always congratulate this because they are fortunate that they have been selected for this faculty training program of this institute, because this is a highly reputed institute, Vishwavidyalaya, because I have seen closely this institute since the beginning of the agriculture department, and they are now flourishing like anything. And many students, they are agriculture graduates, they have been trained at this institute. So I congratulate Dr. Archana and Dr. Vinod Dar and Guru Prasad ji and all these people. They have rightly started this program for these faculty members to enlighten them about the new technologies in agriculture and how they can be utilized in the developing the entrepreneurship and having profit making. At the same time, you must see that a sustainable development of agriculture, so that we must not leave our food self-sufficiency, and now we must have not only food security, along with it, we must have the nutritional security also. So only this biofortified, nowadays, the biofortification of zinc, iron, protein, in all the cereals, pulses and everything, there is a huge program is going on under biofortification. At the same time, recently new technologies have come, the genomating, because now in our Indian conditions, we are not allowing the transgenic crops because of some restrictions. So now a new technology has came like genomating, where they will not incorporate the genes from other species, rather than they will incorporate and try to articulate the genes in such a way it will give a higher yield along with quality and resistance to biotic and abiotic stresses. So now the government is allowing to develop these genomated genotypes of different crops for cultivation. So because it is not having any other gene from other organisms, it is from gene from the same. Suppose if rice is there, they are editing the rice crop, rice genotype for different traits like biotic stresses, like abiotic stresses, high yield, quality, like this. So nowadays, lot of new technologies are coming, and marker assisted selection like the so SNPs and other things. So in that also, the agriculture graduates can show the entrepreneurship in developing a labs for this SNP analysis, genomic selections like that. So I personally congratulate this group of Sri Vaishnav Vidyapit Vishwavidyalaya for successfully conducting this program and enlightening the faculty of different institutions. Thank you very much, Dr. Achanaji.
Thank you, sir. Thank you so much for your kind words and knowledgeable words. And we assure you that we will surely consider all the points and all the suggestions given by you. And, and we will always try to include our students, our BSc, MSc students, and give training to them regarding the entrepreneurship. Thank you, sir. Thank you very much. Now, may I request Professor Vinod Dar, sir, Coordinator, Sri Vaishnav Institute of Agriculture, Indore, to please propose the vote of thanks.
Good morning to everyone. Chief guest on this occasion of the inaugural session of Faculty Development Program, Dr. Sai Prasad, our respected Dean Faculty of Agriculture, Dr. K.N. Guru Prasad, Dr. Ashok Jadhav, Dr. Pandey, who will be the guest speakers of this session. I express my sincere thanks to Dr. Sai Prasad for accepting our invitation to this inaugural session and sparing his valuable time to address our participants. Your lecture was quite enlightening, and I hope the faculty members of the institute, as well as those who have participated in this program, must have felt encouraged and made themselves knowledgeable by your words of wisdom. You have given us thrown so much of light into the agri entrepreneurship, how it can be dealt effectively. About our institute came into existence in the year 2018, and our first batch passed out in July 2022. As we all know, as per the recommendations of the ICAR, the fourth year is totally dedicated to hands-on training. Seventh semester is rural agriculture work experience, and the eighth semester is totally dedicated to hands-on training or experiential learning program. We at this institute have started some modules which are helpful to students for making themselves self-employed or capable of joining the industries like mushroom cultivation, swan production, we have nursery raising, we have seed production, We have vermiculture production. We have also floriculture. So some of poultry also. So we are starting some few more modules which will be helpful to our students to be self-employed when they pass out from this institute. As the faculty development programs are organized for the progression of the career of our faculty members, this year's theme has been devoted to agri entrepreneurship. So what developments have been happening in this area? So accordingly, we organized and invited our guest speakers to speak on such themes so that it could be helpful for our faculty in their career progression, as well as helpful to the students as to serve them as a guidance. So thankful to all the participants, especially to Dr. Sai Prasad sir, for sparing valuable time to address our gatherings. Thank you very much. Over to you, Archana.
Thank you, sir.
Thank you, sir. Thank you very much, and thank you all the members for making this inaugural session so meaningful. Thank you. This inaugural session, it will be followed by lecture sessions, and in this lecture session, we are having two speakers with us today. Dr. Ashok C. Jadhav, a junior mycologist, All India Coordinated Research Project on Mushroom, Pune Center, College of Agriculture, Pune, Maharashtra, and Dr. Pooja Pandey, Director, Founder, Beti Innovatives Private Limited, Indore, MP, with us. So, starting with the lecture sessions, let's have a look over the works which is done by our first eminent speaker, Dr. Ashok C. Jadhav sir. Our first speaker of today's lecture, Dr. Ashok Jadhav sir, is working as a junior mycologist, All India Coordinated Research Project on Mushroom, Pune Center, College of Agriculture, Pune, Maharashtra. He has research and extension experience of more than 29 years. He is also a reviewer of two journals, that is Journal of Agricultural Research and Technology, Pune, and International Journal of Agricultural Science and Bioinformatics publication. He has total 38 scientific publications, 1 book chapter, 16 technical publications, 18 popular articles, 12 radio talks, and more than 4 TV programs. He has also been honored with the Best Performing ICR All India Coordinated Research Project Mushroom Center Award for the year 2019 at Pune Center from Directorate of Mushroom Research, Salon. He is elected as member of Executive Council of Mushroom Society of India by Directorate of Mushroom Research. He has also worked hard to increase the cultivation of oyster and button mushrooms in state of Maharashtra through organization of several trainings, demonstrations, farmers rallies, arrangement of mushroom stalls in different exhibitions, actual site visits, newspaper articles, radios, TV programs, and many more. He has attended more than 3 international and more than 14 national symposium workshops. At present, he is involved in teaching the courses for UG and PG students, and he has guided more than 7 PG students during his teaching experience. So, may I request Dr. Ashok Jadhav sir to please start his today's lecture session.
Okay, thank you, ma'am. Good morning, everybody. I am Dr. Ashok Jadhav, working as junior mycologist at this center. Am I audible, ma'am?
Yes, sir, you are audible.
Okay. So, for this faculty development program on agri entrepreneurship, today's my topic is on mushroom cultivation. Particularly, I will deal with the oyster mushroom and the button mushroom. So, which are grown on large scale in the Maharashtra state and also in Indian state. These two mushrooms, particularly the button mushroom, it is grown on large scale. About 75% of mushroom production of India, it is from the button mushroom. Then it is followed by the second one, that is the oyster mushroom, which contributes about 19% of the mushroom production of India. So regarding mushroom production, of India, the Maharashtra is leading one. As the total mushroom production of India during last year, it was about 1.25 lakh million hectares, million tons, out of which Maharashtra contributed about 30,000 metric tons, followed by the about 500 metric ton of oyster mushroom. So this is the scenario wherein the Maharashtra is leading in mushroom production of India. Regarding our lecture, first I will deal with the oyster mushroom cultivation, the simple technology, and any layman can cultivate this mushroom at its own, and as a entrepreneurship also, he can develop his own business or a livelihood for its cultivation. So I am sharing the PPT. Okay, whether it is visible?
Yes,
sir.
Yes.
Yes,
yes,
sir.
Okay. So, firstly, we will see the cultivation technology of the oyster mushroom. What is the mushroom? So, mushroom is a fungus. The fruiting bodies of the fungus, which are hypogeous or apogeous, and visible to our naked eyes, it is called as the mushroom in simple. And a typical mushroom, it has a cap-like structure, and beneath it, it has the scales or the gills from which the spores can be liberated, and have a stalk-like structure and called as stem or the stalk, and beneath it, the mycelial threads attached with the substrate. So this is the typical structure of the mushroom, which we are observing during, particularly in the monsoon season. We will get the different type of mushrooms in the jungle area or in the nature, wherein the physical conditions like the humidity and the temperature, along with the substrate, available substrate, we will observe the number of mushrooms having different size, different shapes, and different colors, which are to be observed during the monsoon season. However, every mushroom it is not edible. Maximum mushroom, they are poisonous one. Maximum mushroom, they are poisonous, or the edibility, it can't be decided on the spot whether it is edible. But the ethnic information from the tribals, we are doing the survey in the whole region of Maharashtra. We have covered, wherein the ethnic information from the tribals and the some laboratory test, and they can decide whether the mushroom is edible or non-edible. और यू कैन से जिस मशरूम को कीड़ा खा सकता है उसे आदमी खा सकता है, दैट इज द fruiting body in the nature that has been infected with the insect, it may be edible. So but you have to decide its edibility in the lab test only. So this is the typical structure of mushroom. Regarding different type of edible mushrooms which are grown worldwide, more than 16 to 18 different species of mushrooms or types of mushrooms they are grown in the world. We will deal with the first one, the white button mushroom. That is the
Hello,
agaricus.
sir.
Yes, ma'am.
Sir, sorry to interrupt. I think that your presentation is not working.
your slides are not moving.
Yes, sir. Please do check it out.
Achha, achha. I will reshare it. Okay, whether it is visible?
Not yet.
There is a message, your screen sharing is pause.
Your sharing is not visible, sir.
But I'm getting the message, your screen sharing is paused. How it happens?
Dr. Archana, just inquire
Yes,
from
sir,
the
yes, sir.
technical team
Yes,
whether
sir,
they have
I will
paused
check it, sir.
screening.
Okay, sir, I will check.
I think it may be visible.
The slide of morphology of mushroom.
Yeah.
So it's visible.
Okay. Regarding the edible mushrooms grown throughout the worldwide, the first one is the white button mushroom. Whether it is visible now?
It's visible.
Yes, okay. That is the Agaricus biosporus. It is the temperate mushroom and commonly known as white button or European mushroom. It is worldwide grown on large scale, particularly in the world scenario. The shiitake mushroom, it is grown on large scale, followed by the second one, that is the button mushroom. So this mushroom, it requires the different parameters to be controlled while it's growing. So during the vegetative growth, it requires the less temperature around about 22, and at productive state, it requires a minimum temperature of 16 to 18 and the humidity of 85 to 90%. Then the second mushroom, it is the pleurotus, that is the oyster mushroom. It is also known as the wood fungus or dingri mushroom. There are some common species which are commonly grown in Indian states. That is Pleurotus sagaricali, Pleurotus fluida, Eos, then Flagellatus. But this mushroom, it can be.
The slides are not moving.
You may click on the slide number 3, sir.
Sir, you can share your full computer screen instead of sharing this slide. Then it will be movable. I think so.
Is it okay? Whether the slides are moving?
No, sir, not yet. We are not able to.
Hello.
हॅलो सर.
हा
हॅलो
हा
सर, ते तुम्ही जेव्हा स्लाइड ओपन करता ना,
हा
हा तर तिथे स्लाइडला ना ते स्टार्ट स्क्रीन
समझा रही है, इसको समझा रही है। Just scroll to next slides. It's full screen. It's okay. Yes. ये नहीं बोल रहा है अर्चना जी. अगर आप
Okay, sir. Now it's visible.
अच्छा, नेक्स्ट स्लाइड आ रही है.
Yes, sir. Yes, sir.
Okay. So the first agaricus, the second it is the oyster mushroom, that is Pleurotus species. It is also called as wood fungus or dingry mushroom, and these are some common species grown in large scale. So it doesn't require any control conditions, and it can be grown throughout the year. As the temperature requirement for its growth, it is from 20 to 30 degrees centigrade. So throughout the year, this mushroom can be grown throughout the year. Then the next mushroom, it is the milky mushroom, that is Calocybe indica. It is commonly known as Judi chatta, and it is totally milky white in color, and it requires higher temperature and also the higher humidity. That is the temperature of more than 30 to 35 degrees centigrade for its growth and reproductive stage. And it is the common mushroom, and it is grown in the coastal area of Indian states like Kerala, Tamil Nadu, and Karnataka. It is grown on large scale. This is the milky mushroom. Then the paddy straw mushroom, that is the Wolveriana species. It is also called as the Chinese mushroom, and the commonly cultivated species is the Wolveriana wolvacea, and it can be cultivated very easily in any kind of the plant structure like the herb structures. For this, it's growing, and it is harvested within a short period of time. That is within the 15 days, you will get the first harvest for this paddy straw mushroom. And the temperature requirement, it is about the normal temperature of 25 to 30. Then the another mushroom, the medicinal mushroom, the shiitake mushroom, that is Lentinula ilotus, and it is usually grown on wood of dead deciduous trees, and also known as Japanese mushroom. It is very medicinal and nutritional mushroom and popularly grown in the northeastern states of India. But it requires a somewhat less temperature of about 15 to 20 degrees centigrade. In the black ear mushroom, that is auricularia, it is also a wood ear mushroom and it is subtropical and it can be grown on also sawdust or paddy straw or wheat straw. And the temperature it is about 20 to 30. Then the next it is the Ganoderma mushroom. It is the common medicinal mushroom. Ganoderma lucidum is the species which is grown on for its medicinal purpose and it is worldwide popular for its medicinal value. So it is medicinally important. Again, the diseases like cancer, diabetes, then HIV, and also blood pressure. So it is cultivated on sawdust, bran of cereal grain, and it requires some higher temperatures, about 25 to 35. So as I have told, the wild mushrooms, they are observed during the survey. You observe the mushrooms grown on some leaf litter or the dead with treatment. So these are the some wild oyster mushrooms which we have collected from the jungle area of the Konkan area, then Marathwada, Vidarbha, and some jungle area of the Maharashtra. So this as we are dealing with the oyster mushroom, these are some pics of the oyster mushroom observed in the jungle area. So in case of Maharashtra, we are dealing with the mushroom map of Maharashtra by doing the survey for about 12 to 13 years, and of which there are about 131 different species are recorded, wild species, of which 41 are edible, and of which only 4 to 6 species they can be cultivated artificially on different substrates. So these are some wild oyster mushrooms grown on different trees or the dead trunks or the leaf litter. So regarding our today's topic, the cultivation of oyster mushroom. So for growing any crop, you must think about its different varieties. And the varieties commonly grown in India and particularly in Maharashtra are the Pleurotus kaju, which are the mostly acceptable variety. That is the Pleurotus kaju having the grapefruits in the Pleurotus austriatus. It is also called as blue oyster. Initially, the pineal formation is in the blue form, and after at the harvesting stage, it becomes white. It is also a high-yielding variety, Pleurotus austriatus. Then the Pleurotus florida, it is totally white color, and it is growing in bunches like that of the Pleurotus austriatus. Then the pink color mushroom, oyster mushroom, it is Pleurotus eos. It is also growing in the bunches. So regarding the yield and the acceptance by the farmers or the people, the Sajar Kaju, it is mostly accepted by the farmers, followed by the Pleurotus austriatus. As the temperature requirement, it is about 25 to 30, and the humidity, as usual, every mushroom for its cultivation, the humidity it should be more than 80%. So these are the common cultivated oyster species grown in Maharashtra and also in the Indian different states. For its cultivation, the infrastructure requirement will be three parts. That is the spawning or bed filling room. It should be closed, as you have to do the spawning in the some hygienic condition. Then the second one, you will require the incubation room. It should be semi-ventilated for incubation of the mycelium on the substrate. And the cultivation room may be a simple structure with the cross ventilation and provision of the water. So these are the infrastructure, minimum infrastructure required for its cultivation of the oyster mushroom. Regarding the material required, the first one it is the important, that is the spawn, the seed of the mushroom, the spawn. Then the straw, that is the substrate, that is the remnant after the harvest of crop. You can use as a substrate for its cultivation. Different straws you can use, that is wheat straw, paddy straw, soybean straw, cotton straw, or any remnant after the harvest of crop can be used as a substrate for its cultivation. Also, you will require a polythene bag for filling the bales, preferably 35 by 55 centimeters, so that you can prepare a bale of 5 kg. Okay. It should be transparent one, so that the growth of mycelium you can observe from the outside. If for the sterilization of the straw, if you are using different chemical method, you will require the chemicals like biostic and the formaldehyde, and the another one the thread and the mineral. So these are the basic requirements for its cultivation. And the spawn, the straw, polythene bag, the chemical, thread, and the mineral. Uh, Regarding its stage, first one you have to have the spawn. You can procure it, or you can make your own spawn by having a technical training of its preparation of spawn. Then collection and preparation of spawn, then sterilization and the pasteurization of the spawn, then addition of the spawn, that is bed filling, and after care and management and marketing. So the first one is the preparation of the spawn. So it is totally a lab procedure wherein the mother culture they are multiplied on a substrate, particularly the cereal grains, that is like wheat, jowar, or bajra, the mycelium, the multiplied in the lab condition. So these are the different cultures. So the pure cultures you can procure it from the DMR, that is Directorate of Mushroom Research, Ambagat Solar, which is having a gene bank. You can get the pure cultures of the any mushroom which is grown in Indian states in pure form. So these pure cultures are then isolated, multiplied, and maintained in the laboratory under the aseptic condition. So there are some different types of cultures. The pure culture of agaricus, then the oyster mushroom, which are maintained in petri dish or petri plates or water-safe bottles, and maintained in pure form. The mushrooms one, the vegetative mycelial growth of mushroom fungus, artificially grown on the sterilized cereal grains. So this one is the commercial spawn, which is prepared from the master spawn. So this commercial spawn, it is used as a seed. The packing may be of half kg or 1 kg. So for selection of the seed of mushroom, that is the spawn, it should be fresh one. As you can see, the spawn, it is totally pure white mycelial growth on the substrate, that is the cereal grains, without any other contaminating organisms. It should be pure white. But if you are keeping the spawn for a longer period, at old age, due to aging, it may get pigmentation, maybe of yellow stains or pinkish stains. You can observe, particularly in the Florida variety or EOS variety, a small pigmentation you can order, but it is a pure one without any contaminants. For its purity, it should be white in color and without any contaminants. It should be a fully grown mycelium on the substrate. So after preparation or procurement of spawn, the next step it is the preparation of the straw or the substrate for its cultivation. So the oyster mushroom, it can be cultivated on different agro waste or the straws. You can have the locally available agro waste or the straw to minimize the cost of cultivation, so that the cost can be reduced. So the yield of mushroom, it depends on the agro waste or the straw used for the cultivation. So this is the table showing the yield of the sugar variety on 1 kg dry substrate, wherein different straw like wheat straw, paddy straw, banana leaves, sugarcane fans, or soybean straw, or cotton straw, or bajra, any straw you can use. Wherein you can observe the highest yields are obtained on the cotton side, the leaves. But however, as the selection of the straw, you have to go for the locally and easily available straw to be used, so that you will get the minimum cost. Regarding the straw, it should be dried one without any moisture, and it should be chopped one in the size of 2 to 4 centimeter pieces. By the thresher, you can get the straw ready to use, but it should be dry and without any moisture. And the size it should be 2 to 4 centimeters. So for sterilization purpose, you have to get it with moisture for the moisture content. Fill the straw in the porous gunny bag and tie it and soak in the water for about 8 to 10 hours. So that the excess water after draining the excess water, the moisture percent may the moisture percent of the straw may be up to 50 to 60%. So you recover to drain out it for 2 to 3 hours. So first of all, you have to make the straw in top condition of 2 to 4 centimeter size. Then soak it in the water for 8 to 10 hours. And after that, you have to drain out the excess water till the moisture may be about 50 to 60%. So this straw you have to sterilize before it's used. So there are different pasteurization methods. The first one it is the hot water treatment, that is 80 degree for 1 hour, or hot air, that is the steam under pressure, 80 degree for 1 hour. And if you are using the chemical method for the straw sterilization, you can use 7.5 gram biostin plus 125 ml formalin in 100 liter of water. For hot water treatment, first of all, you have to soak out the straw for 6 to 8 hours, drain out the excess water, and keep it in hot water for 80 degree for 1 hour. So this one is the one of the method. Then the steam, you can use the boiler system, wherein you have to maintain the temperature about 70 to 80 for 1 hour. For these two methods, you have to pre-soak this straw for about 6 to 8 hours to give the required quantity of moisture. And you can also go for the organically grown oyster mushroom by using the two methods, that is hot water treatment or hot air. For the steam, you can use the methods for its cultivation. The another method, the chemical method, wherein for 100 liter of water, you have to add 7.5 gram biostin and 125 ml formalin in 100 liter of water. And put the gunny bag in chemical for soaking for about 16 to 18 hours. The container, you can use the plastic drum or the barrel, wherein you have to prepare the solution by mixing these two ingredients as 7.5 gram biofilm and 125 ml formalin for 100 liter of water. Soak the straw for about 16 to 8 hours. You have to take the care to tie the plastic, tie the container or close the container by covering it with the plastic paper. And after that, you have to remove the bag for draining out the excess water, wherein the moisture level may be up to 50 to 60%. So for this method, when you are using the chemical, you can directly soak the dry straw for 16 to 18 hours in the solution of biostin and formalin, and after that you have to drain out the excess water. Then the next step, it is the formation bed filling. So the bed should be filled in a closed room. As I have told, if the bed filling room, it should be separate one, and it should be closed room. Under aseptic condition, you have to do the spawn. That is the addition of the spawn in the sterilized substrate. So the sterilization of polybag, it is must, as you are using the polybag, and you have to sterilize it with the 2% formalin, the polybag, your hand, and the place of bed filling by using 2% formalin. So rinse the bag in 2% formalin solution, and it can be surface sterilized, and it is used for the bed preparation. Then you have to go for the spawn. Before using the spawn, you have to check its purity. So it should be free from any contaminant. It should be totally pure white mycelial group on the substrate, and it should be free from any contaminants. Before using, you have to confirm its purity. Then take the polybag, invert it at the base, and make the add a small or little quantity of spawn. You have to add the spawn by layer method in the sterilized straw by layer method. As the base, as you are to taking the mushroom all over the bed, while after removing the bed, you will get the mushroom from all over the bed. So at the base, you have to add the spawn, little quantity of spawn, then add the sterilized straw, having the moisture percentage of over 50 to 60%, a layer of sterilizer, and then again add the spawn, then the sterilized spawn, again the spawn. That is the layer spawning you have to do, and the spawn it should be used at the rate of 2% of the wet sterilized straw. Suppose you are preparing a bed of 5 kg, the spawn rate it will be 100. So for preparing a bed of 5 kg wet sterilized straw, you will require the 100 kg seed, that is the spawn. So from 1 kg seed, you will prepare a bed, 10 beds of 5 kg. So during its straw sterilization, the weight it will be increased by threefold. If you are soaking a straw or sterilizing the straw by any of the method, the dry weight it will be thrice. Suppose you are sterilizing 1 kg, the final weight of the straw will be 3 kg. So accordingly, you have to manage for its quantity, then the quantity of the spawn. If you are having 1 kg sieve, you will require about 17 to 18 kg of dry straw to prepare 10 bags of 5 kg wet straw. So the spawn it should be added at the rate of 2%. After adding the spawn by layer method in the sterilized straw, you have to tie it with the thread. Tie the bag with the thread, and with the help of pin or a needle, you have to make the pinholes all over the bag for its aeration or the growth of the mycelium within the straw. So with the help of pin or the needle, you have to make the pinholes and keep this bag for the incubation. So after addition of the spawn, you have to keep the bag at normal room temperature for incubation, that is the growth of the mycelium within the substrate. So within the 18 to 20 days, the whole bed it will be covered with the white mycelium of the mushroom fungus like this. So during incubation, the temperature it should be around about 20 to 30. Okay. So in case of oyster, the incubation period is about 18 to 20 days. According to season, it may vary from 15 to 22 days also. So the whole substrate will be covered with the whitish mycelium growth of the fungus within 20 to 18 to 20 days. And these bags are ready for the fruiting like this. So after incubation, completion of the incubation period, you have to remove the plastic bag after the complete spawn run, and the bags are kept on the nylon hangers, or you can have the iron racks also for keeping the bags for fruiting. So after opening the bag, you should take care of watering once or twice in a day as per the environmental condition to maintain the bed condition as a moist during its whole tenure. So watering should be a regular one after opening the bag. So during incubation, you doesn't require any water, but you have to maintain the semi-ventilation during the incubation period so that the mycelium will grow fast within the substrate. So after incubation, you have to just clear out the bag, keep it on the hangers, or you can use the bamboo racks or iron rack and go on watering. So after 2 to maximum 5 days, you will observe the initiation of primordial. That is the pinhead formation in there. Small mushroom will appear from the surface that is prepared bed, and the base after will appear after removing the bag. So the mushroom it requires the opening for its growth. So after clearing the bag, you go on watering, and the third or fifth day, you will get the small pea head, and after that, within 3 to 4 days, you will have the fully grown mushrooms which are ready for harvest. So the fruit should be harvested at its maximum size and before curling of the outer edges. These are the signs for its harvesting. The curling of outer edges and the maximum fruit size. This is the indication for its harvesting stage. So the first harvest in case of estimation, it will be within 20 to 25th day or maximum 26th day. The mushroom is ready for harvest. So you have to just harvest all the mushroom fruiting bodies on the bed by twisting the fruiting body clockwise or anti-clockwise. Don't pull out the fruiting body that will, as it will disturb the bed. So the fruits are harvested by twisting clockwise or anti-clockwise. After the first harvest, suppose it may be at 24th or 25th day, harvest all the fruiting bodies, go on, remove the other parts of the straw, cut it with the knife and go for market or pack. After the first harvest, you have to just scrape out the bed with gentle hand for and keep the bed for re-incubation. So from the same bed, you will have the second flush like this. In the first harvest, at the first flush, around about 50% of the yield you will get from the single bed. After 8 to 10 days from the first harvesting, the second flush will be ready for the harvest. Wherein the number of fruits and the size of fruits also will be small or less, and the second flush you will get about 35% of the yield of the mushrooms, fresh mushrooms. Like that, you can also have the third flush from the same bed. Okay, it will be of 8 to or maximum 12 days, 8 to 12 days from the second harvest. The third flush, it will be ready for harvesting from the same bed, wherein the number of fruits are also less, and about the remaining 15% of the total yield will be opting from the third harvest. So these are the harvesting we can take from the same bed. Three harvesting you can have from the same bed. So after harvesting the mushroom, you have to just remove the basis of the straw, relative the fruiting body, then make the packing. You can use the phonics or the attractive packing in the polybag for marketing. So this is the simple cultivation technology, wherein any layman can have its cultivation, as it doesn't require any specific structures or any. You can use the huts also or the poultry sheds wherein you can grow for the oyster mushroom. The structure it should be covered, okay, by side way and the roof way. So or you can see different farms where the low cost technology most of the farmers are having for its cultivation. So this one will be quality mushroom farm from the Satara district, wherein the oyster mushroom is grown on large scale. The nylon thread hangers they are commonly used as it is cost effective at low cost. You can prepare the beds or keep the beds on the hangers. So this will be farm. Then the another farm from the Raigad district of Maharashtra. So the small farmers are engaged in this cultivation. Then some self-help groups or the Mahila Bachat Ghar, they are also growing the mushroom small scale or medium scale or some large scale also. Or in their own houses, in a closed room with the ventilation facility, they can grow the oyster mushroom in their rooms. So this one will be mushroom production unit from the Yerwada jail from Pune. When the hangar system it is being used, we then got the boiler system for it, the straw sterilization. So this is the you can have the proof with the pin or PC sheets like this. You can prepare the racks with bamboos or the nilgiri from Nashik, wherein the bamboo racks are used for its cultivation. Similar from it is from Pune, the hangar system. These are the structures where it is covered with the green sheets, and the sides are covered with the green shade, so that you can have the ventilation. For the farmer, they are using the green shade. So after harvesting, you have to go for packing. When packing, you can also from estimation, it can be dried and stored for about 6 months to 1 year in dry condition. So 1/10 volume is reduced. So if you are drying 10 kg of estimation, you will get 1 kg of dry estimation. So the connect packing or iron drum. These are the poultry cells which are converted into the mushroom culture, oyster cultivation, wherein the fogger system or the rack system, they are using grading and the pattern. Okay. These are the structures which are available in the condition for its cultivation. The hangar system. Regarding the oyster mushroom, most of the farmers or the oyster growers, they are near about Pune, about in Maharashtra, about around about 475 oyster growers are there. And the total oyster production of Maharashtra it is about 460 metric ton per year. So the major crop wherein the small entrepreneurs or the agri entrepreneurs can go for its cultivation without less investment. Within in 10 to 15,000 investment, the farmers can go for its cultivation. As it doesn't require any specific conditions or the specific structure, so you can use the huts or any available structure. It should be closed one with the ventilation facility, so that in low cost technology, this oyster machine can be grown. So this is regarding the oyster machine. Now I will go to the another machine that is the button machine. So regarding button mushroom, it is totally grown under controlled conditions. So the investment in its cultivation is very high, and as we say, the economical feasibility of project it depends on its cost of cultivation. So in case of button mushroom in Maharashtra condition, it should be totally grown under controlled conditions, because you have to manage all the parameters like temperature, the humidity, or the comfort preparation and the spawn production. So in Maharashtra is leading in India for button mushroom production. There are about 39 big projects are there having different production capacity. The biggest one it is at Baramati in near Pune, having a production capacity of 20 metric ton per day. It is followed by the Mane Agro, the big project at Talegaon near Pune, having production capacity of 12 to 13 tons per day. Then the another third one is by the Wakefield company, having production capacity of 8 to 9 tons per day. So this mushroom it should be started on a large scale. It cannot be cultivated under a small condition or a small scale. So the minimum feasibility, you can say the economical viability of buttermilk, it depends on its production capacity. So as economically, you can say it should have the production capacity of minimum 1 ton per day. Then and then the button machine will be economical feasible or economical viable. because you have to manage all its parameters, cultivation parameters. It is the tedious and technical and skillful. So to manage all these parameters, you will require the automation, mechanization, then some machinery and equipment, and the land requirement will be around about 3 to 4 acres. So the first aspect in case of its cultivation, it is the compost preparation. Okay. So regarding compost preparation, it is technically a very skillful job to prepare the compost. The major aspect in case of button mushroom project cultivation, it is the compost preparation. So for the, as we know, the agriculture biospheric is temperate motion and requires a different condition at different stages. It will require about 22 to 28 degree temperature during spawn run and phase run, while at fruit formation stage, it will typically require a lower temperature of 15 to 17 degrees centigrade. And as usual, the humidity from the growing one, it should be maintained at about or more than 80 to 85, and enough ventilation facility during its propagation. These are the typical requirements for its cultivation. And there are three basic steps for its cultivation: the production, the procurement of the spawn, as we have studied, then the production of compost, and the third one is cultivation, wherein the spawning, casing, and grazing the crop. The infrastructure or the material required or the equipments required for its cultivation are the equipments like air conditioner, exhaust fan, then steam boiler, composter, humidifier, weighing balance, humidity, then the material like the straw, agriculture waste, then spawn, the FIM, fire feed, there are some minor material, then the chemical and the infrastructure. Regarding infrastructure, you will require a separate composter, then the separate pasteurization unit, phase 1 and phase 2, then the separate spawning room and insulated cropping room and the boiler. The major aspect, it is the compost preparation. It is the identified as a microbial degradation of the organic waste of the substrate. The white button mushroom, it requires the will decompose method, as it is heterotrophic or separate fungi. The biological activity of number of microorganisms make the substrate selective for its agar species. So degradation of organic material that is destroyed, which are converted to the soluble form of nitrogen into the microbial substances. So the compost, it is very selective or specific in nature and should be properly prepared, which can support the growth of the aggregates. The raw material are, there are different formulas are there for its preparation of the compost for the button mushroom. The material required for its composing, the first one is the different straws or the cereal straw, wheat, paddy, or most of the people they are using wheat straw. Then the sugarcane bagasse, paddy straw, maize straw, soybean straw, in straw. Then and these straws are the reservoir of the silver, semi-silver, lignin, and it should provide the carbon nutrition. And it should be freshly harvested at 8 centimeter, shiny in color, and the nitrogen percent it should be from 0.4 to 1%. If the CN ratio of the material is high, it has to be supplemented with the nitrogen soap to bring it down for its fermentation. Or some of the farmers they are using some supplements like horse manure or chicken manure or sheep manure or some supplementary carbohydrates to balance the CN ratio. Some farmers they are using some nitrogenous fertilizers like urea, calcium ammonium nitrate, or ammonium sulfate. The nitrogen content of these are very high, which is released quickly and resulting in quick establishment of the microflora. Some concentrate means the animal means or wheat or rice bran. There are some concentrated meals are used, and this material they can supply both nitrogen and carbohydrate, which is in case of the animal mammals are released slowly. So the nitrogen content of the animal food it is about 3 to 12%. Supplements like muriate of potash, superphosphate, gypsum, calcium carbonate. Commonly, the gypsum it is exclusively used as a supplement, which can balance the pH. Also, it helps to remove the glycemic, and it's also the sustainable colloidal material. So these are some nitrogen contents of raw material, which for horse manure, then chicken manure, wheat bran, soybean. These are the nitrogen percent and the moisture. So nitrogen computation should be there. It should be around about 1.75 to 2.1, the initial nitrogen of the different stock or the ingredients. The elements, nitrogen and some other elements are there also. You have to manage or calculate. Different formulations. The final compost should have the CN ratio of 30 as 1, then the nitrogen 1.5 to 1.75. Then the final CN ratio it should be 16 to 70 as 1, and the color it should be dark brown and it should not be fizzy or sticky. And the final compost should have the pH of neutral, that is 7.2 to 7.8, with the moisture of 60 to or maybe 65 to 60%. There should not be visible growth of undesirable organism except
Dr. Ashok, excuse me.
Yes, sir.
Your slides are not moving.
Not moving?
No. Make it full, your presentation full screen, and then scroll to the next slides. Make it full screen.
Whether it is visible, sir?
It's visible. The first slide is visible. Let you try the subsequent slides, sir, to check whether those slides are moving or not.
Yes, I am moving it. Whether moving, slides are moving?
No.
No.
Sir, you can just click on slide share button so that it will be visible to everyone.
अभी दिख रहा है? Hello?
Sir, but the slides are not moving. We can see the presentation. Slides are not moving when you are presenting the data.
No, it's no, they are moving, but make it full screen, sir. Make the slides full screen. Make it full screen.
Yeah.
Sir, third option, sir. Go to third option. It will be easier.
Yeah. It's at full screen now. Yes. Whether it is moving?
No, sir.
No.
Now it's full screen.
Yes, sir.
Yeah.
Now it's
We
full
can
screen.
see.
You can just scroll to the, yes,
Yes,
now it's all right.
it's all right. No.
Yeah.
Okay. Then I will again start with the brief one. For the agaricus, it requires a specific temperature, then humidity, different steps are there, cultivation, then production, and the compost preparation. So the first one, it is the important parameter, it is the compost preparation, for which we require for its cultivation, different equipment, material, chemical, or the infrastructure uh, for growing, then for compost preparation. So compost, it is the infinite microbial degradation of the organic waste, and it should be a selective one for the agriculture species. The raw materials like agricultural waste, then the animal waste, or the nitrogenous fertilizer or concentrate meals, they are used for its preparation, and there are different formulas for its preparation. And different supplements like murat of potash, then gypsum, calcium carbonate, these are commonly used as a supplement for the compost preparation. But the gypsum, it is commonly used to have the pH balance milk, then removing the greasiness, and it serves as a calcium nutrition for the agricultural biospheres. So these are some nitrogen content. Before we have to calculate the ion content of the different raw material, it should be 1. In between 1.5 to 1.75. So the elementary requirements. The formulation, it should be designed so that the final compost should have the CN ratio of 30 as 1, then nitrogen at 1.5 to 1.75, and final CN ratio, it should be 16 to 17 at 1. And the color of the finished compost, it should be dark brown with the pH of 7 and neutral pH with moisture of 65 to 68%. So there should not be any visible or undesirable organism except the actinomyces or the free of insected waste. So these are different formulations. Some formulations suggested by the DMR Solar, but at a large scale production, different growers, they are having their own formulations with different small changes in the ingredients. With the commercial farms in Maharashtra, and this is a common formula. Wherein the Vista, suppose you are taking 100,000 kg Vista, the poultry manure at 50%, that is 500 kg, then you have 15 kg, with brand 70 kg, and the gypsum around 35 to 40 kg. This is the common formula which the farmers are using. Instead of Vista, most of the farmers, they are using the sugarcane bajaj, and soybean straw, or paddy straw, or the another straw. So there are two methods for its preparation, the long method and the short method. Most of the farmers in North Indian states, they are following the long method of compost preparation, uh, like Punjab, Haryana, then Bihar, wherein they are the seasonal growers for the button mushroom. They are availing the natural facility during the winter, so that they can have the lower temperature, and they are mostly using the long method of composting. So by long method of composting, the compost will be ready within 28 days, or it may take about a month for its preparation. So these are common ingredients, whereas in fish straw, then these are some ingredients used for the long method. We store in calcium nitrate, murat of potash, superphosphate, urea, glucose, or the gypsum. These are the shapes wherein the farmers are preparing piles. For the flow chart, in case of long method, you have to first wet the ingredients and the straw, mix them, and the moisture, it should be about 70 to 75%. Then prepare the pile to keep for 1 to 5 days. Then after that, you have to go for first turning, then the second turning, the third turning, Likewise, you have to go for 7 to 8 turning at the interval of 3 to 5 days and break down the pipe, and after that the compost is ready within 20 to 30 days, ready for I can make the heap and pour it and remove the sheet and check and compost and add the spot. There are the structures, low cost composting shape, wherein the compost piles are prepared uh, for the long method of compost. Or on the open spaces, these are the piles prepared by mixing the different ingredients. These are the commonly method following North Indian states. Mix the ingredients related with water, add different ingredients. The moisture level which should be more than 75 to 72 to 75%. Also the ingredient chicken manure or gypsum, mix it by addition of water. You can have the machinery like buckets or the front end loaders for mixing turning the compost. Also, and make a pile. This is the cross section of the compost pile, wherein the aerobic zone is there. When the trial ranges, they will obtain have the growth in the semi-arid zone and the anaerobic zone in the middle of the pile. The mechanization we can use the pile makers or with the help of the laborers we can prepare the piles. So the shortcoming of there for the long method, dry matter loss is there. Between sprays of insecticides on the side, most of the amino acids lost in the atmosphere. So you will get the lowness, it have the whole smell during its preparation. So it is not equal environment trained. The another method, most of the farmers, they are using button machine to grown under control condition. They are following the short method of composting. So it consists of two phases. That is the outdoor composting, that is phase one, which is of 10 to 12 days, wherein the 90% chemical process and 10% biological process. The phase two, that is pasteurization and conditioning of the compost inside the tank under the different set of conditions, that is 90% biological oxidation and 10% chemical process. For short method, pre-wheating area, pasteurization, staking board, blower, ventilation, humidity probe, thermometers, ammonia, oxygen meters, and boilers. These are some machinery required for preparation. The common formula, that is viscose, wheat bran, chicken manure, urea, and the gypsum. So the typical scheme, the structure of button machine for minimum 14 to 16 rooms should be there with the spawning area, then the manure shed, straw shed, then the compost yard, and the phase one and phase two for the compost preparation. So that we will get the 1 ton machine production per day. For short method, for phase one, mixing for chicken manure ingredient and making and staking, then break down the pipe, add water, then again restake it, then add other ingredients and make a aerobic step. Then the second day, first turning is given, then the second turning after 2 days, then the third turning, and after fourth turning, you may shift this compost in phase two. So in this method, totally chemical process is there, 90% and 10% biological processing. In case of phase two, we'll transfer in phase two. pre-pasteurization conditioning at 45 to 52 degrees centigrade. Then chilling is there, and pasteurization at 59 degrees centigrade for 4 to 6 hours. Then post-pasteurization at 45 to 48 degrees centigrade. Then post-pasteurization, cool down the conditioning at 25 degrees, and then go for the spawning. So all these parameters, they have to be managed with the automation, mechanization, and the sensor, along with some machinery like equipment and air blowers. So the purpose of pasteurization conditioning, it reduces the time of composting, and it converts the ammonia into microbial protein. So most of which otherwise goes waste in long method of composting. Then it kills the or inactivate some insect, pests, or diseases or competitor modes during its process, and conditioning of the compost. Then it increases the biomass of the thermophilic microorganisms, which favor the growth of agricultural species. So in short method of composting, more compost per unit area can be obtained. Characteristics of phase 1 and phase 2, it should be brownish when properly hydrated, that is moisture about 72 to 72, will give smell of ammonia and pH about 8.2 to 8.5. The acting masses are not so apparent. So these are the structures. Phase 1 provided with the air blowers. This one is schematic diagram. The air blowers are provided wherein the compost is using. You have to manipulate the airflow with the VFD, air handling units, of the chambers, okay, where to fill the compost. Or you can have the conveyor method also. On a large, big project, they are using this mechanization or bobcat machines or the front end loaders. Digital thermometers, then the sensors, they have the records of temperature requirement at different stages at different times. Sorry. Floors, multiple floors. That at the phase 1 and phase 2. So from phase 2, it should be decomposed to the dark brown, soft. The moisture percentage should be about 65%. It should have pleasant smell, no thickness, stickiness, and when content it is the 2.1 to 2.2. Ammonia it is below 10. This is the flow chart of phase 1 and phase 2. Here the parameters like temperature can be CM ratio we have to manage for the calculation of the influence and the some machineries like blowers. So in case of bulk pasteurization, more compost per unit you can obtain. The cost of pasteurization is less, and the yield per unit paid for competitive high. Another method it is indoor method, wherein the compost can be prepared within a short period of time, 12 to 14 days. The compost it is ready for spawning. This is the schedule, wherein within 12 to 14 days the compost it is ready. These are the structures. Phase 1 and phase 2. The phase 1 it is within the piles. Like the phase 1 it is for construction. Like the floors. So indoor composting, it doesn't need any costly machinery. The compost yard it is equal to 1/3, no emission of foul smell, and cost again the labor can be reduced, raw material can be used. there are some disadvantages of the indoor composting. So low bulk density of the composting water, and the look, that is brownish look of the composting is not good. That is two attributes. The final compost is to be brown in color. Nitrogen level as I have told, it should be 1.5 to 1.75. Moisture up to 65. Then it will now decrease in pH at neutral and without any crystal disease. So in brief, by long method, short method, and indoor method, you will require a month for long method, in short method up to 16 to 18 days, and by indoor method within 12 days, the compost is ready. Selectivity is partial, it is control, and it is also control. The average yield is 10 to 15 kg per 100 kg of compost. In short method, somewhat higher yields are often in indoor method, down to 30 kg per 100 kg. Regarding the environment, it is long method is totally polluting, it is moderately polluting, and the indoor method is non-polluting. And the average compost production per ton of the stock is 1.75 to 2. It is 2 to 2.5 in short method, and in case of indoor method, it is mostly triple, 2 to 3.5. And the average N content 1.75 to 2. In case of short method, it is 2 to 2, and in indoor method, it is 2.2 to 2.5. The infrastructure required for 20 tons outdoor compost yard, and covered compost yard in case of short method, and the waiting area in case of the indoor method. Land is required for it is somewhat higher in long method, then followed by soil testing the indoor method. The power requirement for long method doesn't require any machinery or equipment. In case of cover in short method, you will require power equipment. It doesn't require for long method. In case of short method, you will turn on fire filling machine and hot water production machine. In case of indoor method, filling land, then hopper, bobcat, or JCB. So these are the basic requirement for the preparation of selective compost suitable for the agricultural species. So it is a very tedious method for preparing a compost for the button mushroom. Then regarding its management of the button mushroom, the crop management, the another aspect in case of button mushroom. Whether it is visible crop management.
Yes, sir. Yes, sir.
Okay.
Yes, sir. We can see.
Okay, after a tedious method for compost preparation, you have to go for the crop management for the button mushroom production. So in case of agronomical crops, the management is very easier. But in case of button mushroom, you have to manage all the environmental parameters. So the spawning, then the casing, the cropping, watering, harvesting, and the post-harvesting. These are the some management practices to be done. So after preparation of compost, you have to go for the spawning, that is addition of the spawn in the prepared or the finished compost. So the spawning of the compost, it is done immediately after the completion of compost process, and when the compost temperature that is cooling down, conditioning at 25 degrees centigrade. What is spawning? It is the process of mixing the spawn in the compost of the final compost. This is only spawning. There are different methods, and the surface spawning, the layer spawning, or door of mixing spawning. Most in the commercial farm, door of mixing spawning is used. The rate of spawning, as in case of oyster mushroom, it is 2% of the wet stock, but in case of button mushroom, it is about 0.75 to 1% of fresh compost. The spawning rate it is of 1%. In generally 1% of spawn it will keep. So the low rate of spawning it will slow down the spawn rate or the spore growth of the mycelium. While the high rate of spawning it will raise the temperature and can kill some of the spawn. So the spawning it should be a proper level, minimum 0.75 to 1% of the finished compost. So after preparation, final compost, which will be brownish in color without any contaminant, and have a good smell, and should not be sticky one. This one you can add manually or by mixing. Then after spawning, you have to fill the bag and place the bag in proper rooms. So filling of 10 kg spawn bag, it is the common practice followed in most of the farms, wherein 10 kg compost bag is prepared. The bags are placed in the shell or the racks arranged in cropping rooms. Then the spawn run, that is the growth of the mycelium within the compost. It's called as spawn run. And the temperature requirement during the spawner stage, it should be around about 22 to 24. And the relative humidity of that room, it should be more than 80, and the higher concentration of CO2, that is more than 1000 ppm. And the spawner, that is the growth of mycelium on the in the compost, it will take about 10 to 12 days. This one, after filling the bag, you have to keep the bag for the spawner. Like this, tie the bag and keep the bag in the iron racks or the shell for incubation, and that is growth of mycelium and the finished compost. Like this. So during this stage, you have to maintain the temperature at about 22 to 24 degrees, so that it will favor the growth of mycelium to grow within the finished compost. Then after that, the casing. So after the spawn run, you have to go for the casing. So it is the covering of the spawn run compost with a layer of pasteurized soil, casing soil, or any available material. So this casing metal it should provide the physical support to the growing machine, and it also helps to regulate the flow of nutrients from the compost to the developing machine protein body, and it also acts as a reservoir of water. It also facilitates the food body development. So it is the essential step for the fruiting in the buttermilk. For making the casing material, different materials are used, different formulations are there. In the old days, maximum FOM was used as a ingredient for preparation of the casing. So the mixture of 2-year-old FOM and 2-year-old spent mushroom compost at one as one is used, or another formulation of garden soil plus sand as 4 as 1, or decomposed FOM plus soil as 1 as 1. But the common method during the last few years, the common method, it is prepared, the casing soil, it is prepared with the help of coco peat and soil as 80 as 20%. So, the casing soil prepared by the prepared with the coco peat 80% and 20% garden soil. Before using this casing material, you have to go for it sterilization, and the sterilization can be done with the help of steam pasteurization. That is steam at 65 to 70 degrees for 6 to 8 hours, or by the use of formalin 40% at the rate of 2 liters in 40 liters of water per square per cubic meter. These two methods can be used for its sterilization by addition of calcium carbonate to manage the pH of the casing material. Like 80% soil, then 20% coco. For the mixture, there disseminated on a small scale, but in large scale, there are the chambers wherein the ingredients are mixed in proper proportion and applied with the steam under pressure for about 6 to 8 hours. The formalin 4% will be applied within the chambers and the casing material to sterilize before it's used. So the qualities of the casing soil, the good casing soil, it should have high water holding capacity, it should have the high porosity, it should be light in weight, and it should be nutritionally pure, and the pH it should be near about neutral, and it should be well distributed. So after preparation of the casing method, you have to apply it on the spawn bag, and that is the spawn completion of the spawn. So level the spawn run comfort bag by breaking the top 2 to 3 inch spawn run comfort and press it slightly. Apply 4 to 5 centimeter thick layer of casing side over it and level it, water it, and maintain the temperature at 22 to 24 degrees centigrade along with 90% humidity. So this case run, and that is the growth of the mycelium within the casing layer, is called as case run, and it will take about 8 to 10 days from the casing. So this is the fully grown compost, spawn run compost, wherein the you have to apply for the creation of casing material. So before that, you have to prepare the casing material with suitable ingredients like cocoa plus soil, sterilize it, and apply the 2 to 3 centimeter layer on the spawn bag. And this level it and go on water. The water should be minimum so that the layer of casing up to the layer of casing it should be weighted. And the data kept in the racks, and during the case run, the temperature it should be about 22 to 24. Required to after case run, the cropping. So 1 week after casing, the root temperature should be brought down to the 15 to 17 degrees centigrade. Then and then there will be putting of the formation of the primary. And the relative humidity it will be maintained about 80 to 85 degrees, along with the good ventilation at this stage for its putting. Then the CO2 level it should be below 21%. To be provided with fresh air, and the clean air starts happening within 3 to 5 days of the fresh air injection and lowering the temperature. So after case done, you have to suddenly drop down the temperature to 15 to 16 or 17 degrees centigrade along with the fresh air, so that the cleaning formation will be start within 3 to 5 days. Then go on regular water like that. These are the cleaner formation, wherein you have to maintain the temperature at 16 degrees centigrade. This is forming. At this stage, the temperature requirement is about 15 to 16 degrees. Along with lower concentration of CO2, these are the cleaner formation. Then after cleaner formation, you have to go for harvesting. So the cleaner appears within 10 to 14 days of the casing. The peanuts mature within 4 to 5 days, and the mature mushroom they are harvested at the button stage, that is 4 to 5 centimeters cap diameter. And the cropping continues for about a month or 4 to 8 days. And the mushroom appears in the flushes after every 7 to 10 days. And mostly the farmers for the growth they are having, or they are taking 2 or maximum 3 flushes they are taking. After that, they are vacating the homes. The average in Indian condition it is 15 to 25 kg in 1 to 1.5 months. So from 100 kg compost, you should get about 20 to 25 kg of fresh button mushroom. This is the yield. So these are the size of mushroom ready for harvest. Or some of the farmers, they are using the shelf method, wherein the tray method they are using. The compost spawn with the spawning, and they are put for the incubation, wherein the temperature is maintained. This is another method instead of bag method. But in this method, the risk is there. If there is attack of different any disease, the whole crop failure will be there. Instead, in bag method, if there is appearance of any disease within a bag, you can describe it or describe a particular bag, so that we can avoid the contamination to the other bag. So this is the flow chart. After the preparation compost, then compost preparation, then spawning. After spawning, you have to go for casing material preparation. Then the spawn run is there, then the casing, then the case run, then pinhead formation and harvesting. So during spawn run, case run, then pinhead formation and harvesting, you will manage the parameters, then the hygiene condition, and then the control of any diseases of the pest. So this is the flow chart. Okay, in brief, it is narrative regarding the cultivation of these two mushrooms, that is the oyster one and the butter. This is all about from my side, sir.
Thank you, Dr. Ashok. Dr. Archana. Dr. Archana. Dr. Archana, are you there? Yeah. Dr. Archana, carry on.
Hello.
Yeah.
Thank you so much, sir. Thank you so much for your such a deep and knowledgeable lecture to us. And I hope that every participant, he must be updated with the knowledge of mushroom cultivation technologies, whichever you have shown and you have presented today. So along with cultivation technologies, it also become important that whatever mushroom which we have cultivated, when, where, and how it should be sold, how it should be marketed for getting a proper price. And for such reason, we are having our second guest lecture, which will be delivered by Dr. Pooja Pandey ma'am, and she will be sharing her experience and her knowledge regarding the marketing strategies and entrepreneurship, all the strategies regarding the mushroom. So before starting the lecture, let's have a look of her work. So Dr. Pooja Pandey, she is the director, founder, Betty Innovatives Private Limited, Indore, MP. She is working on agri waste to well solving malnutrition and pollution problems. She has developed the technologies to replace thermocol using agri waste. She has developed various value-added products from mushroom and superfoods. She is involved in tissue culture and manufacturing quality mushroom seeds under strict laboratory conditions and mushroom productions. And she has conducted a professional business-oriented training on mushroom cultivation and tissue culture techniques. She is working on various environment and malnutrition practice, and she has also attended the various workshops, training programs, exhibitions, and along with all this, she is also having her teaching experience of more than 5 years. She has published her work in more than 6 research papers. She has also attended various national international conferences and presented poster oral presentations along with the abstract submission. She has also been honored with various awards and prizes for her work. Her success story is also been covered by Ashtak team on 30th October 2022. She has given training to Israel and US clients on tissue culture and mushroom cultivation techniques in August 2022. Along with such a brief profile of Dr. Pooja Pandey ma'am, I request ma'am to please deliver her lecture on the topic entitled Mushroom Entrepreneurship, a Sustainable Future. Ma'am, please.
Thank you, ma'am, and very good afternoon to each and everyone present here. Am I audible?
Yes, ma'am.
Yes, sure.
Okay, thank you. Kindly allow me to share the screen first. Hope it is visible.
Yes, ma'am, it's visible.
Okay, fine. Thank you. Thank you for the wonderful introduction, Dr. Archana ma'am, and I'm really thankful to Sri Vaishnav University for inviting me and providing me this wonderful opportunity. And also I want to pay my regards to all the eminent speakers and the panelists present over here. And it was really a great experience to hear Dr. Jadhav with such deep insights in mushroom cultivation, specifically on oyster mushroom and button mushroom, with thorough knowledge of practical part. Especially, I think most of the people would have got a great inspiration and knowledge from his session. So myself, Dr. Pooja Pandey, I'm the founder of Beti Innovative Private Limited. And today, I think I should make it a full screen first.
Yes, ma'am.
Yeah. So my company name is Beti Innovative Private Limited, and Beti stands for Biotech Era Transforming India. And I started basically in 2017 with my mushroom spawn production laboratory. Before that, I was working in Tata Cancer Hospital, Mumbai, and I'm also the Young Scientist Awardee from Madhya Pradesh government 2014. And I was awarded, and then I got the fellowship, and I went to National Institute of Virology, Pune. And after that, I joined Tata Cancer Hospital, Mumbai. And then I thought of starting something after coming back to my hometown. From my knowledge, from my research experience, whatever I have gained in throughout my life. And as this session is for faculty development, and also I think some students would also be present here around. So I would like to tell them that they should use all their knowledge, and whatever they are learning, everything is hidden in books. Anything, whatever you are reading from your courses is going to lead to entrepreneurship. And today's session is on mushroom entrepreneurship, on which I'm going to deliver some information and my experiences, whatever I have gained. So mushroom entrepreneurship could be a sustainable future. And when we talk about entrepreneurship, it doesn't mean always a business. It means like you are also doing a business along with some social work. And when we talk about sustainability, it definitely, because the whole world is talking and moving towards a sustainable environment, sustainable future. So our company Beti is working in that direction only. And in 2019, our company got this registered under Startup India, and it is a DPIIT certified startup now. And we have also received a grant from central government. That is a great motivation to go forward. So what are the current issues? We see that we dream of a beautiful India, wonderful India, with a full greenery around, the beautiful houses, the healthy air to breathe in. But we know that how is the condition of Delhi. The parali issues are there every day in the newspapers. As the winter is coming, at the time we see the fog issues because of the paralis and the agri waste being burnt. And we see the climate change is a great problem in our country, and not only in our country, the whole world. Water crisis is a challenge for the future. Land is getting degrading. It is also getting desertified. And burning agri waste, as I told, is a major issue which is creating a lot of pollution, and carbon emission is there. And another issue is malnutrition, unemployment. So these are the issues which are like we must say we should focus on when we think about entrepreneurship. And when we say entrepreneurship, mushroom stands perfectly fine. The slides are not changing. It's changing now.
Yes.
Yeah. And the major deficiency in our diet, this is a very big issue, as I told, malnutrition. The major deficiencies are vitamin D, vitamin B12, vitamin A, and protein. So high prevalence of hypervitaminosis D is being increasingly recognized globally. So we should understand and we should focus on the problems when we think about any entrepreneurship, any business, anything. Not only it should be for generating revenue, it should also help our society directly or indirectly. So mushroom has proved itself, and it has proved itself to be a superhero for this, I must say. As we see the 17 sustainable goals of UN, which are going to lead us to the creator to solve the problems which I addressed just now on the screen, the problems like poverty, zero hunger, and these all like 17 goals are being targeted by UN, so that we can sort out we can lead our life in a peaceful and a healthy country. So mushrooms, being wide biotechnology, they are really very much helpful in helping us out in solving out these, mainly the 17 sustainable goals which UN has raised to, and it has to be like it was raised in 2015, and by 2030 it is being considered to sort out the problems which are sustaining around the social issues, the environmental issues, the malnutrition problems, the pollution problems. So we see that fungus is a solution for all these kind of problems. And you can see fungus comes in the agriculture, it comes in the biotechnology, it comes in all the industries. So anything we can choose, if we want to enter in entrepreneurship, any one of the field we can choose, and here fungus and mushrooms can help us out in creating entrepreneurship also and sorting out some problem. When we talk about the dream of startups and the startup India, what has started, it really means that you should have some innovative idea, you should have some you should be addressing some problem, you should be solving some problem which is sustaining around. So from a single solution you can address a number of problems around which which we can see in our day-to-day life. So first, we should be knowing what are fungus and how they came, like people came to know about all these things and all. We should be knowing that mushrooms are basically fungus, and fungus, they came on the earth before the plants itself. As I told, that carbon dioxide is a major problem, the global warming, the climate changing issues. So the whole world is thinking how to reduce the carbon emission level in this world. So mushrooms are fungus, and we should like it has already been published in many research papers through the phylogenetic and protein sequence analysis. That is a little bit biotechnology part, but through sequencing and all, it has been proven that around 1,000 million years ago fungi were present on our earth. That is before the plants, because plants came around 700 million years ago. So fungi were present, and when the earth was made, at that time, the plants and fungi were the most important living being which were helping out the earth to cool down and reduce the carbon dioxide. And we have forgotten about the fungus and mushrooms. We know about the plants, but we are having very less and limited knowledge and information about the fungi, which are playing a secret role in the nature which we are not knowing. That is, they are in the forest, in the untouched areas, they are naturally working to fix the carbon dioxide in the nature. So mushrooms, we see, is a very important future for us to grow mushrooms and to eat mushrooms. Both is very important. So now let us see that what is the status, how many fungi or mushrooms are present on earth currently. So around 1.5 million. You can see 1.5 million fungi are estimated here on the earth, but out of that around 110,000 are fungi which are reported already, which are like the taxonomist, and it has been classified under the literature, scientific literatures. Out of those 140,000 are considered to be mushrooms, but still only this 14,000 mushrooms are reported. Only 14,000 mushrooms are reported till date. Out of those 14,000 mushrooms, 3,000 are the prime edible mushrooms, which we know that it can be eaten. It has been proved through the different methods of testing, through toxicology testing and through physical appearance, and finally it has been confirmed. So out of these 3,000 prime edible mushrooms all over the world, I'm talking about the whole world, not because in India we are very much lacking in these data and all. Out of those 200 are being cultivated experimentally, but in India only 6 are produced on industrial scale. I will focus more on this 6 in the next slides, coming up slides also. So mushrooms are basically basidiomycetes. Mushrooms are basically basidiomycetes, and few are ascomycetes also. But mostly whatever mushrooms we see, we eat, we have like grown are mostly classified under basidiomycetes. Some mushrooms, just I will tell you, will come to know, like cordyceps mushrooms and morels. These are the mushrooms which are ascomycetes. Rest of the mushrooms are basically basidiomycetes. So mushrooms, I told that it is actually playing its role in nature automatically. It is helping the sequestration of carbon. Besides that, you can see here, it is also acting as a www network of forest. Like we say World Wide Web, here it is Wood Wide Web. So under the earth, beneath the earth, it is connecting, transferring the communication, creating the communication between the trees, and exchanging the information through molecules and some chemical releases, and they are helping out the trees to communicate between each other. This has been recently studied. These are very interesting findings, which now as these days people have found interest in fungi studies and all. That's why these all data is generated. Even recently some publication, some videos were there which were showing the sounds of the fungi, the fungus, the mycelium, which is interacting with the plants and within themselves also. So it is creating a kind of sounds. It has been studied recently. Some videos were published on YouTube and all. So fungi are very useful for us. They eat death and create new life, because we don't know. I told that they are playing their role secretly and unknowingly. We are not knowing about mushrooms. Many people are not knowing about mushrooms, and but they are directly, indirectly playing their role, just like the vultures and all in the nature. They clean up the environment. Whatever CO2 is released by us, we are generating a lot of pollution. They are fixing it. The whatever waste biomass we are generating, they are fixing that also. So they are actually eating the death. That means they are the real decomposers. Whatever farming is going on in our lands and fields, whatever waste is generated, if it is not decomposed by the fungi, then it will become a big pile in a long time accumulation. So this is a very important, because without fungi to aid in decomposition, all life in the forest would soon be buried under a mountain of dead plant matter. So fungi are the garbage disposal agents of the natural world, and they break down dead organic matter, and by doing that they release nutrients, and those nutrients are then made available for the plants to carry on growing. It's how everything is reborn. So that is, this entire web of life is connected, you can see. So this is the scientific classification of fungi. I told that it is most of the mushrooms are whatever we eat and we see around under Basidiomycota, and the kingdom is fungi. The subkingdom is Dicaria, and mushrooms are basically mainly mushrooms, rust, and smut. These three comes under Basidiomycota, and Ascomycota consists of morels, truffles, brewer's yeast, and baker's yeast. So the bread we eat. Well, nowadays what happens, like when we talk about mushrooms, when I started my mushroom entrepreneurship, at that time many people were not aware about mushrooms. So when I was talking or giving any awareness session, creating any session and lectures, at that time people are saying that we don't consume mushrooms. They are actually they consider it either a non-veg food. So we have to explain them that mushrooms are basically fungus, and you are already eating breads, you are eating already this idlis, dosas. These all are fermented foods and they all contain fungus. So baker's yeast, brewer's yeast, you are already using these type of microbes in your diet daily, but still people are not aware about the mushroom. So there is a lot of need to create a lot of awareness among the people regarding especially the vegetarian and non-vegetarian difference. Some people consider it as a vegan food completely, although it has a separate kingdom in itself, fungi, and it was earlier considered that it is a plant. Later some of the characteristics match with the animal kingdom also, so they were separately kept in a different kingdom, that is the fungi kingdom. So we need to educate people around us wherever we go, wherever we meet, because then only people will start eating mushrooms. Because in China is the leading producer of the world, and we are just 2% of that. The whole production and consumption is done by China only. So we see that mushrooms are basically, we can see they are either edible, either medicinal, either toxic and poisonous. So when we talk about mushroom, what is the first thing comes in our mind? That chhatri type image, that umbrella type of image. We think, okay, when I'll talk to any customer or any layman, they will I will talk say them that this is edible mushroom. They say what edible means, we don't know. We have seen the kukurmutas and all. They talk such kind of things. Like we should explain to them, and they should be knowing that the mushrooms are just like plants. We see the grasses growing around, but we don't consume them in our daily life. But still we know that each and every plant growing around us is useful, or maybe useful for some medicinal purpose, edible purpose, or some other applications. So all the things applies to mushrooms also. Mushrooms are basically not able to prepare their own food. As I showed in my previous slide, they are decomposers. That means they are saprophytic in nature. That means they don't prepare their own food, and also they don't kill any living being to prepare their food. They will just feed on the dead organic matter, and they will grow from that waste material which we are creating around us. So we should consider this a very important part of our life, not only for eating purpose and for other applications also. Just like the plants, we give importance to plants, we understand the importance of herbs, we have so much respect for plants, we know that this this herb is useful. So mushroom is also a kind of herb. It should be given respect because it is playing a very important role in our nature, and we must say that it is not only healing us, it is also healing the whole planet. So whatever mushrooms we see around, maybe it may be edible, it may be medicinal, or toxic or poisonous. But when we say mushroom is toxic or poisonous, so here comes again a myth that people are afraid of eating mushrooms, that it could be, some people will say it is non-veg food, some people will say it is toxic food. So here we should understand this thing, that many plants, many fruits are also toxic. We know when we will go to a forest and we see any unknown tree, we will be afraid of eating that fruit. Maybe it is looking like a grape, it is looking like a guava, but we will be afraid of eating it because we don't know that whether it is toxic or safe for us. So same thing is applied for mushrooms also. It could be either toxic also, and it could be edible also. The consumer should have a proper knowledge, or else he should purchase it from the market or should grow himself or herself. Then it is a safer concern. If you don't know, if you don't have idea, otherwise you have to study about it, get a proper knowledge. Many people, we must say many tribal people, still they are, many people are in my touch, those who are taking spawn from us. They are saying that every year in September we get the natural mushrooms in the places we stay around that area. We harvest those natural wild mushrooms and we eat it. It is really safe. So I asked them, how do you know? They say there are some traditional techniques to find out whether the mushroom is poisonous or not. And once they miss, then we can see the news in the paper next day if it is missed out. So you should be aware while consuming mushrooms. I'm saying mushrooms are healthy. Then it doesn't mean that we should go to any wild place and harvest mushroom and consume it. It is not like that. It is just like plants only. So edible, medicinal, and toxic and poisonous. So mushrooms are saprophytic in nature. That is, it feeds on the dead organic matter, whatever is present in the nature, and it extracts, it converts the whatever waste is present, and it converts it into a very healthy edible form or a medicinal form. So I think most of us would have seen this recently. Sir had, Dr. Jadhav had showed in his lecture the Ganoderma mushrooms. So whenever I show in any presentation, many people, those who have visited any village area in the during the rainy season or any wild places, they always raise their hand. Yes, I have seen this. I have seen on a mango tree or any old cut tree. So I think most of you would have seen this medicinal tree. Like this season, during the rainy season, I had collected so many Ganoderma during my foraging visits and all of mushrooms. So if in future it happens, if it is possible, I can show in future. So see what is the scenario of our country and the whole world. See, we have current production of around 0.23 metric tons. Out of that, around button stands for 70%, and tropical and subtropical less than 30%. Tropical and subtropical mushrooms means the remaining mushrooms, like oyster mushrooms are had shown in his slides, and remaining varieties are also there. I will show in the further slides also, like shiitake is there. Some exotic varieties are there besides oyster, shiitake, paddy straw, milky. So these all come under tropical and subtropical category. So they account for only less than 30%. So the whole world production is 40 metric tons, and we are doing only 0.23 metric ton production. And what is the scene in our country? We have crop residues in India around 700 metric tons. And I recently told you that mushroom grown agricultural waste, mushroom grown organic waste. So that waste is just going waste, and it is creating just pollution by burning. So let us all get together and collect all the waste and grow mushrooms out of that and create a very healthy, super nutritious food. It is a real superhero. So residues utilized are only 0.06%. You see, there are 700 metric tons, and out of that only 0.06% being utilized. And what is our consumption status? We consume only 0.15 kg mushrooms per capita per person, and the whole world is eating 5 kg. So see, we have to increase this ratio just by creating awareness, making people understand the value and importance of mushrooms. So here is our strength. This is our strength. We are varying in agroclimatic conditions. We can grow seasonal varieties of mushrooms. Who says you invest a lot? No need of investing a lot on mushrooms. I know that button mushroom, you may be knowing that button mushroom growing needs a lot of investment, definitely. But there are some techniques developed by our eminent scientists of India, DMR. They have developed some technologies. From that, you can either grow seasonal mushrooms mushrooms, button mushrooms also, and rest of the varieties you can grow according to the season. And we have huge agriculture waste. We have very highly variable agroclimatic conditions to select the type of mushrooms, and then huge agriculture waste. We have cheap labor. You know, China don't have that much of labor. They work on the machines with the help of machines mostly, and they always say, if I would have the type of labor in just like India, we would have grown very far. So we have cheap labor, we have people to work here, and we have wide biodiversity also. We have many natural varieties of mushrooms which are still unexplored. People out of our country are going to the forest in the seasons, they are doing foraging, they are doing mushroom hunting, and we are not in the rainy season, we are sitting at home waiting for the rains to get finished. Why to go in the deforest? So there is a huge market if explored, not only in India and out of India. China is the leading producer, leading exporter, and it has covered the whole market, and we are only 2% out of that. See, these are some of the few varieties of mushrooms which I am showing you on the screen. I think if students are there, they would love to see this one mushroom, Amanita muscaria, that is fly agaric. And this is the iconic mushroom. Whenever we say the kids to draw a mushroom, they will definitely draw this one in all the cartoons. My daughter loves watching mushroom cartoons and all. So whatever cartoons are there, the kids are really getting attracted specifically towards this Amanita muscaria. And let me tell you that this is a poisonous mushroom, as we discussed the varieties, types of mushroom, edible, poisonous, and medicinal. So this one is currently categorized. This one, I'm talking about this one, red and white, Amanita muscaria. So this one is currently categorized under the poisonous mushrooms. But you know that when we say for plants also, many plants and fruits are poisonous, but we always say, even if it is not edible, there is some other application which could be very useful for us, for the nature. So I'm a biotechnologist basically, so I think always in that direction, that mushrooms are also a very important part of biotechnology. And this mushroom is currently being used as a biopesticide. You understand? Natural pesticides can be created from this mushroom. It kills harmful microbes, harmful insects and reptiles and all. Besides that, not only kids, the whole world, scientists are like after this mushroom, running after this mushroom, because one more variety is there, Amanita phalloides, besides Amanita muscaria. Both are very poisonous, and this Amanita muscaria is also currently known as magic mushroom, because it has the chemicals which we consider them toxic. The lower dose of it has been found very useful for the depression patients, for the people those who are facing depression problems. The minor dose is being standardized and found to be very useful. So you can search on the web, on the YouTube and all. When you type magic mushrooms, then you will come to know a lot of information. The world is running after this mushroom, at which we consider it as a toxic mushroom, and we just neglect it. And you see here, this left one, I told, in the fields, when you go into the wild forest, you will be able to see this Ganoderma lucidium. This is so beautiful, attractive mushroom with such a shiny waxy texture, and it is very useful mushroom. It is not directly edible, but it is one of the important medicinal mushroom, because it is useful for cancer treatment and many other diseases. Especially, it is a great immunity booster. People are nowadays interested in drinking Gano tea, and there are a few companies. See, these are all the things I'm telling you, because each and every mushroom itself is an entrepreneurship for future. So if you take one mushroom, you can start an entrepreneurship from that, your own venture. So there is a company named DXN. If you search on the web, you will come to know that that company is working only and only on this mushroom specifically. This from this Ganoderma mushroom, they have developed a number of products from head to toe. They are solving problem out of that. They have created the shampoos, they have created the face packs and everything, even Ganoderma tea. Even you may be aware about the Vestige company. They have launched the capsules of Ganoderma. So I had some visitors in my place. They said, why you have written here Ganoderma on your board somewhere? So they said that I'm from Vestige company and we have a product that is the capsule of Ganoderma, and we sell it for immunity boosting. So this is one of the opportunity for you all. And here you see cordyceps. This is the booming, it is really booming nowadays. Cordyceps militaris and Cordyceps sinensis, there are different varieties in this one also, but Cordyceps militaris is currently being grown easily in the laboratory. We have also standardized the technique on almost all the mushrooms whatever are being cultivated in India. So Cordyceps militaris is grown in liquid media initially, and then some media specifically required, which has to be standardized. And then it can, earlier it was it is considered basically a parasitic mushroom because it is in Hindi known as Kida Jadi. So it attacks on some insects and on the higher mountain, and it was earlier when it was not cultivated in the laboratory, it was considered that it is very difficult to grow this mushroom. So only the people, they call in the China, they used to call it Yarsa Gumba, and they climb on the very high mountains and they harvest this kind of mushrooms, some sinensis varieties, and they are they're selling on the rate of gold. It was considered as a gold. So it is very expensive nowadays. Also it is most expensive also. Here Morchella esculenta, this is Morel's mushroom. This is also very famous mushroom. Recently only in Myanmar, the technology for growing this particular mushroom has been standardized. Earlier it was only stated and claimed by China that they are having the production, they are able to grow this mushroom. But recently it is proud to say that our country is also able to grow this mushroom now. And this is Lanternula idodes. It is really a super mushroom. This is shiitake mushroom. This mushroom also you can grow very easily, although the production efficiency is less, but it can be grown on the just like the method sir has shown in the for the oyster mushroom cultivation. The difference is only that it is specifically, it needs a compatible environment. Specifically it can be grown on the wood dust, sawdust, and also some species, some strains are there which are developed by DMR, which are also grown on straw, wheat straw, but they produce less amount of mushrooms, but still they can be grown on straw also. So but what is the importance of this mushroom? This is a very super nutritious mushroom. One strain is released from DMR which is considered to be having a very high content of protein, all the vitamins, minerals, even I must state add on here that I had, as I had shown in the previous slide, that vitamin D. So all the mushrooms have the capability to prepare vitamin D, but it can be enhanced through UV exposure and some other standardized technology. So this is a mushroom which can be sold in the powder form also. If the powder is consumed, it will help you out in maintaining all the daily dietary requirements for your body. And this one is Hericium erinaceus. This is very famous in like I had few clients recently, ma'am has told, from Israel. They said it is really famous in their country, Hericium erinaceus. This is हिंदी में इसको शेर की दाढ़ी या बंदर की दाढ़ी, monkeys mane, lions mane के नाम से भी जाना जाता है. और इसको देख के तो लगता नहीं है कि this is edible. It doesn't look like कि ये खाने योग्य होगा. But this is really an edible mushroom. This is lion's mane, and it is being used in soups and various dishes. And also the medicinal properties also there of this mushroom. It is helpful for Alzheimer's type of disease and all the neurological issues which we are facing nowadays in our country also. So scientists and all the researchers are focusing towards the development of this mushroom and selling of this mushroom, mainly out of India, because here in India people are only knowing button mushrooms. So Yeah. So as I told that what are the commercially cultivated mushrooms in India. In the previous slide I had shown you, I wanted to give a picture in your mind that this is the future. These all are the possibilities for us which we can go forward. Because this is an unending field. Once you enter into this field, you will never stop. You will always be innovating. You keep on innovating. It will never end. You will always be coming with a new idea, new thing, because I will show you that how much wide this field is. So so 1, 2, 3, 4, 5. I've shown you here 5 varieties: white button mushroom, Agaricus bisporus, oyster mushroom, this is Pleurotus species, milky mushroom. Am I audible now?
Yes, you are.
Yes, sir. Yes, ma'am.
Thank you. Milky mushroom, this is Psilocybe indica. This is paddy straw mushroom, Volvella volvaceae, and this is shiitake. I had shown you in the picture previous slide that but you may not be aware about this. Mainly people know white button mushroom. This is Agaricus bisporus, this one. We go to the malls, when you go to a hotel, when you will order a dish, the person will give you the button mushroom dish only. They will, if you will ask to them that what variety of mushroom you are serving, they won't be aware about it. They will simply say that we know only mushroom, we don't know anything else about mushroom. So when you will order, you will get in the malls, hotels, everywhere, white button mushroom. This is Agaricus bisporus species. But nowadays some people, especially Dr. Jadhav sir is from Maharashtra, I think. So I have seen, I was in Pune when I was in NIV, I have seen on the small stalls and everywhere, people are very well aware in Maharashtra area about this oyster mushroom. But in our area, Madhya Pradesh, no one knows in the market about this variety. This is Pleurotus species. There are around 10 to 12 varieties in this. This is the only mushroom in which there are so many varieties. Different type of colors are available. Sir had shown in the picture in his presentation. So when I went to the market in Pune, they said, I asked them, do you have any, they were selling the mushroom paratha. So I asked what mushroom you have stuffed inside. So they said, yes, it is oyster mushroom. So I asked them whether you know which type of oyster mushroom you have stuffed inside. They said, yes, it is Sajar Kaju. I was so happy to know that people are so much aware in that area about the varieties even of oyster mushrooms. And here comes milky mushroom, Psilocybe indica. This is mostly grown in South India, but for our condition also, like what we have experienced, theoretically it is said that from March to September you can grow, but we have a few bags which are still giving milky mushroom production. So we can say, because this year season got a little bit changed and it was raining till last month also.
It can be grown in the subtropicals also.
Yes, sir. Yes, sir. It
Yes.
can be grown. Yes, sir. Subtropicals also. Yes, and even in this region in Indore also, we started some bags in the March, and it was still giving. It is still giving fruiting until October. Also, it is giving fruiting because the temperature has not raised that much. Although the temperature range for growing milky mushroom is generally 25, not 25. It is for spawn run, but it is around 28 to 38 degree mainly. But at this temperature also, in this month also, we are able to get it because the conditions are nowadays favorable for this. So basically this is the summer variety milky mushroom. Otherwise white button mushroom, if we are planning for a seasonal cultivation, then
Yes, we need to make cultivators aware of these types of mushrooms.
Yeah.
Otherwise, only two mushrooms are grown:
Yes.
white button mushroom and oyster mushroom.
Yes, right, sir.
And the
Yes.
rest of the mushrooms they don't cultivate.
Yes, right. They are not aware about it.
Because of the lack of awareness. Yes.
Lack of awareness. So we can grow it, sir. We have done all these varieties here also, and I think Dr. Jadhav is the very experienced person here. We are having. He is having so much of experience in technology development and mushroom growing also. So, but we here also, last year we grew shiitake, paddy straw. We are not growing here because, as the name suggests, paddy straw grows specifically on paddy straw. Yani जो पराली होता है हमारा, उस पे ही उगता है mainly. So here, like how to decide which mushroom to grow? There are two major factors to decide. First is the climatic conditions where you are staying, and the second is the type of agri waste you are producing in your place. So these are the two major deciding factors which helps us to finalize which is the best mushroom economically as per our conditions to grow. So if we, like I said, milky mushrooms are mostly grown in South India because they are having that kind of temperature conditions most of the times. And paddy straw, it is mostly grown in West Bengal, Odisha, this kind of area because it needs a high humidity. Besides paddy straw specifically, it grows on that only. So there people eat a lot of rice and they have seed.
In Jammu and Kashmir state, we grow white button mushroom, oyster mushroom,
Yes.
milky mushrooms,
Yes.
and paddy straw mushroom also.
Yes, as per the climatic conditions and agri waste present.
And you have models that is, but they are in wild. We have not
Yeah.
yet been
Yes.
able to cultivate or domesticate it.
Yes, correct, sir. Correct. It like technology has not come to the ground level yet.
Right.
It
We
is.
need to just pay attention.
Yeah.
Just domesticating these models.
Correct.
They are really novel.
Yes.
Of the region.
Yes. Yes. You are from that
Novelty.
area.
They are novelty.
You are from that area, sir.
Yeah, that's right.
Okay. Okay. So that's why you are so much aware. Okay, great. So shiitake mushroom, although what I said that like seasonal mushrooms we should prefer, but if we are getting a good market for a particular variety, then it is better to maintain the climatic conditions artificially. We should say it as a control unit, and then you should go ahead because nowadays what people are doing, because the market preference is for button mushroom, so it is being cultivated in the commercial control units. So this is the way to enter in this industry. Either you choose as per your the climatic conditions and agri waste, or you decide as per the market in your area, market requirement. So here it is shiitake mushroom, you can see. This is paddy straw mushrooms, and this is milky mushroom. Okay, and this is white button mushrooms, and this is oyster mushroom. So some more like this is such a wide field that if we keep on studying for 5 to 10 days, then also it will be very less for us. So these are the different species of pleurotus mushrooms. Yeah, I told you that oyster mushroom has a wide varieties, around 10 to 12 varieties are there, but these are the varieties which are commonly cultivated in India. You can see here, this is Pleurotus florida, the white one. This is eos mushroom, or there are two varieties in this one, jammer and eos in pink one, and this is sajar kaju, and it is known as Indian grey oyster mushroom, and this is Hypsizagus almerius. So there are blue oyster mushroom varieties are also present, and besides this is Pleurotus citinopleurotus. This is a yellow variety. It is very rarely grown in India. It is mainly out of India it is cultivated. So these are the few colors, and people ask us when we provide them the spawn, they ask that do you add the color to this spawn, then they become colorful. Like so I what I have to explain in their language that you grow the roses and you put the green plant always, but do you add any color to it, it will automatically give you a pink, red, white, yellow flower. So the same thing is here also. So this is the way, like I told, that we are lacking in the lands, water, and the unemployment is there. We are lacking, we are having the land getting desertified, and there is a lot of agri waste. So this is a great solution for all kind of problems, and the answer is mushrooms. Because hello.
No, please carry on.
Yes, ma'am.
Yes. Okay, so the answer is oyster mushrooms. Here, this is the pink variety we grew. You can see these plates. These are also developed by us. This is the whole after mushroom cultivation. Whatever, like we grow, what kind of flow we have made. We utilize the agri waste for mushroom cultivation. And once mushrooms are harvested, we develop value-added products out of it. And once value-added products are developed, whatever waste is left behind from the mushroom cultivation, we never leave anything waste. We utilize that also to develop this kind of plates and all many materials we have developed. We can I will show you in the coming slides. So during the COVID time, the mushrooms got some attention from the government. Like they released a poster from their website of my government that in the list of boosting immunity foods. So mushrooms was the first one in the list of vegetables. You can see I have highlighted here. So like now government is also understanding, and like since the time of COVID, people have also started the understanding the importance of mushrooms, and that is the reason this mushroom is getting into a good future. We are going to have a good future of mushrooms, I think so. And people are a little bit more aware because they call us and ask about the details of the mushrooms. So this is the nutritious profile. Mushrooms are not only delicious, they are nutritious also. And this is the health benefits of oyster mushroom, because oyster mushrooms specifically, when we talk about mushrooms, now we say that they are superfoods, they are full of nutrients, they are having protein, iron, vitamin, calcium, vitamin D, everything. But each and every mushroom we talked about have a specific biomolecule which can lead to a industry in itself. So out of India or in our country also, people are consuming some statin capsules. Those who are having cholesterol problems, BP problems, they are eating this kind of medicine, statin, lovastatin. So oyster mushroom is a natural source for this. It is the biomolecule which is specifically present in a higher concentration as compared to other varieties of mushrooms. Oyster mushroom is having this statin and lovastatin. Rest of the mushrooms like cordyceps is containing cordycepin. Different mushrooms have different biomolecules of specific importance, although the basic features are similar. But we say na 19-20 wala difference. So here that 19-20 mein protein, iron, calcium mein 19-20 hota hai. But there is specific biomolecule present in each and every mushroom. So they are found to treat a particular kind of or control a particular kind of diseases. That is why I told you about that lion's mane mushroom. It is useful. It is known as a brain mushroom. Oyster mushroom is good for cholesterol and all. And it is also, there are so many research papers published to control diabetes, as beta glucan polysaccharide is present in this. It helps in preventing cancer also. It is not a treatment, it is a prevention. When people get some diseases, then they call us that now we are having deficiency of this. Once you are deficient, it is not a medicine. It is a food which prevents you from leading to any diseases. So don't consider mushroom as when there are some specific studies. There are a lot of studies going on animals, but human studies are very less to state on a particular type of disease to treat. It could be, but we cannot say it verbally just like that. So it is better to include mushroom in your daily diet so that you may not be having this kind of problems in future. So it helps in preventing cancer because it is having vitamin B12 and selenium. And I told shiitake mushroom, it is having a very good combination of vitamin B12 and vitamin D both. And vitamin D is also present in mushrooms. It is not naturally present. Well, just like our skin, when it is exposed with the sunlight, our skin is having cholesterol that is getting converted into vitamin D3. Mushrooms, when exposed to sunlight, that is the major gill part I will show you in the coming slide. When it is exposed to sunlight, it will prepare vitamin D for you. So it is not having, but it is preparing vitamin D for you, and it will help in boosting your immunity and especially in absorption of your calcium. You would have seen in the ad of Boost or Horlicks, I'm not remembering exactly. They in their advertisement, they said the mother was asking to her friend, "आप दूध तो दे रहे हैं, पर इसका कैल्शियम इसको मिलेगा कैसे?" So they were telling, we have come up with a fortified formula of vitamin D in this product. So vitamin D helps in absorption of calcium. This is very important vitamin for all of us. Since COVID time, people have understood the importance of vitamin D because it also helps in boosting your immunity. So it is a very important vitamin. It is containing good quantity of potassium, which helps in reducing blood pressure. Vitamin B2 is also present. It helps in stimulating absorption of iron. Low fat and high fiber. So this is we must say delight of diabetics also. Those who are having diabetes, they are told not to eat a fatty food, and they need to have a healthy food like protein diet and with high fiber. So mushroom is a very good solution for them. So this is my daughter, Ira, and there were some studies which I found that they were stating, there were some research papers specifically for Plutus florida and Plutus sajur kaju mushrooms, saying that they have anti-carcinogenic and anti-inflammatory action of Plutus florida and sajur kaju, the two common Indian varieties of oyster mushrooms. So this is about recycling. Once we grow mushroom, we harvest it, we consume it either fresh, either in the dry form, sun-dried form, either in the powder form, or through value addition, whatever way we consume. After that, whatever waste is left out, that is known as spent mushroom substrate, SMS. We say it spent mushroom substrate. Substrate means food on which mushroom was grown. That food was agriculture waste. Either it could be wheat straw, that all sir has explained in the previous session. So I don't think I need to explain, but we know that all these things come under agriculture waste. From that we do mushroom cultivation, and whatever residue is left behind after mushroom cultivation is known as spent mushroom substrate, and it has many other applications, not only one single application. It can be used as a fodder for animals. It can be used to improve the health of the soil, like whatever bags which we are putting for our spawn testing and all, we are simply using that SMS in our pots, garden pots, and they are giving very good results. Like if I have put a tomato plant in that, it is enhancing the yield of it. So you can do such kind of trials and scale it up. This is also one of the part of could be a future entrepreneurship. Fodder, whatever waste is generated, but remember, if you have grown mushroom from chemical method, as sir had explained in this presentation, through Beverstein and formalin method, then never use it for fodder. If you have used grown the mushroom through organic means, either through stream treatment of your straw or through the hot water boiling method, any of the cold pasteurization method, in that case you can use it for as a fodder, because it could be harmful for the animals if any residues are there of the chemicals. So you can standardize a nice fodder for the animals. It is a demanding market nowadays. Then bioremediation, treating the waste, we must say xenobiotic or recalcitrant waste that is not naturally degraded in nature, that can be naturally degraded by the mushroom waste also. So if you put a bag of waste like SMS in the water which is dirty, and if it will pass through it, whatever most of the big impurities, small nanoform impurities are there, it will be degraded through that mycelium present in that bag, through the fungus present in that bag of mushroom. So it will help in bioremediation also. Organic farming can be done with the help of waste generated from this. It can also be used for biogas production. Mushroom cultivation, again, in case of button mushroom, some people are using oyster mushroom waste, and also for vermicomposting. So it is a mixed venture. You can start from this. These are the future prospects of waste also, whatever waste is generated from mushroom. So this is mycelium in health. I told mycelium word I have not explained to you. Mycelium is the fine threads, the structure, the roots of the mushroom which spread around. You can see the white threads whenever any fungus you would have seen. So that is known as mycelium, and that mycelium is very rich. The lower you go, like if you go on the top of mushroom fruit body, there will be less protein. As you go downwards, you go toward the stalk part, there will be more protein. When you go towards the mycelium part, there will be more protein in that. So nowadays we are focusing in the mushroom production, dehydrating it, making powder out of it. But out of India, people have gone far away. They are not, they are growing mushrooms, but some people, those who are into some, we must say, into the biotechnology industry or some fermentation technology, they are using solid state fermentation technology, and they are directly growing the fungus, the mycelium of mushroom, and making powder of it and extracting the important biomolecules, which I told each and every mushroom is having a specific biomolecule of commercial importance. So So these all, you can see the list: beta glucan, lentinan, cordycepin, ergosterol, ganoderic acid. These all are extracted directly from the mycelium powder, or they are using it in a dietary supplement, as a functional food, and adjuncts in the drug formulation. So this is the future of mushrooms. You can see these are the each and everything I'm telling you is itself a future prospect for entrepreneurship. This is also like some industries in India also now entering in this field. This is myco meat, we can say vegan meat, directly growing mycelium on the plant proteins and all, and they are creating this kind of meat alternatives. You can see this is the electro microscopical studies of this. This is spun soya, chicken, and mycoprotein. You can see the fibers around those people who are addicted of eating non-veg and they want to leave it, but they are finding it difficult. This is a very good alternative for them. So this is the future. This is the future. Mycomaterials, we are also working on this. You can develop the different kind of biodegradable articles. Leather can be made out of it, and this many biomaterials can be developed, any kind of packaging materials and all. And even I have also shown you in the previous slide. This is the future. Pharmaceuticals application, like cosmetics. Nowadays there's a mushroom, snow mushroom. From that, people are preparing creams. For they have extracted the nanoparticles out of it, and they have developed the creams which help in making people fair more. So this is applied in cosmetic industry, nutrition industry, and you can see the nutraceutical, nutri-cosmetics industry, food industry, beauty industry. You can start anything out of that. You see here mycelium based materials are being developed because it is a naturally growing thing now, so it can be grown on agri only any organic waste. So you can develop things out of it. You can grow the materials on your own. You can see there are some companies, IKEA and all, they are developing these kind of sustainable articles. When we say sustainable, we mean that the industry or any entrepreneurship which you are starting should be using such kind of raw materials which are either renewable or not leading any pollution to your nature, and not utilizing all the naturally present things, so that you may not leave anything for your future generations. So it should be a sustainable thing. Your entrepreneurship should, or the government is also now focusing towards the sustainable industries, sustainable things. So, and people should also start thinking about the future of their coming generations, because the thing is that if we will use everything, if we will develop we are working in an industry and we are utilizing the materials which are not renewable and which are also leading pollution, then what will be the future? With what we will end up? That will not be a good just earning money. So mushrooms can heal people through health and as well as wealth, because it has high nutritional value, medicinal value, probiotic in nature. And I don't think I need to explain all these points. Probiotics and all, good for diabetics, weight loss, immunity booster, but also for wealth. It creates self-employment. It is a low cost production and more income. Low illness rate is there. SMS, I told, it has also good applications to generate money out of that spent mushroom substrate, and utilizing agriculture waste, leading to decreasing the pollution and malnutrition problems. So mushroom can heal people, mushroom can heal the planet. These all are the future prospects from mushroom. Mycoremediation, mycopesticide, I told you the poisonous mushrooms can be used for developing biopesticides. So everywhere the myco terms come. When fungus is there, everywhere myco comes. Mycoremediation, bioremediation becomes mycoremediation, biopesticides become mycopesticide. Mycofiltration, you can filter your water. Myco restoration, you can regenerate the forest. I told you in the beginning that they help to connect the the communication of the trees in the wild. And myco adaptation, thermocol replacement, biocoal replacement. So it has a great future. and that too sustainable. So Beti's vision, our company Beti's vision is to upscale traditions scientifically. So whatever things which we have shown you in the slides, which we are out of those, we are trying to innovate one by one on every aspect we are working. So our mission is currently to eradicate malnutrition and pollution from our country. Yeah. Hello.
Yeah, ma'am, you can continue. Sorry.
Okay. So I would like all to join our hands together to eradicate malnutrition and pollution from our country. So these are the images from our lab. You can see the spawn which we prepare. I started with this work. Spawn, we do tissue culture, and these are the objectives to grow, because if we want to grow this industry, we should have a good quality of seeds. That is known as spawn technically. So spawn is the vegetative mycelium part which is grown on the wheat or any other grain which is supportive, but we generally work on wheat in India mostly, and it is sterilized, and then it is autoclaved. After that it is inoculated after maintaining the pH and checking all the parameters and all in the under the laminar sterile conditions. We used to inoculate with the culture, specific culture of specific mushroom. So this way we are working. We also provide dissertation and research projects to students. There are some BAVV and girls from Vanasthali and Jaipur University. And this is Beti mushroom spawn of milky mushroom, oyster mushrooms. You can see here. Just this is a video. I think I have to escape, then only I can show. This is a video, I think, just to show you in brief how tissue culture is done, and we prepare the spawn. This is the media tube. The culture is prepared. This is the mycelium. This is the mother spawn. Then this is the final mushroom spawn. We grow mushroom like this also. So this is the spawn we prepare in our laboratory. This was just to give you a brief. So recently, these are the visitors from Israel who came to learn from us many technologies and all. And also one more idea I got from them, that one more future entrepreneurship prospects are there, mushroom tourism. They are interested in creating mushroom tourism in India with other countries. So this could be a good future for us, for our country also. And this is my daughter Beti Ira. And this is, I told that the technology which this is ZDPT method will be used for button mushroom cultivation. This is the mushroom cultivated by us. This is button mushroom, which is cultivated from this technique, ZDPT method. This is oyster mushroom of Beti. And these all are varieties. We last year we grew all these things in one place, in a small area. Button mushroom, oyster mushrooms, and milky, not milky, oyster mushroom varieties. Milky in summers only we start. This is the way, like currently this was the issue that people are packaging either in the thermocol or in the plastic based punnet packaging. So we worked on the packaging also. This is dry mushrooms. This is the powder of mushroom powder. You see that this is the gill part. This I was telling, this is the gill part of mushroom. When it is exposed with the sunlight or UV light, it prepares vitamin D for us. It is a natural source of vitamin D2 for us, and it is a vegan vitamin D. So once it is dried in sunlight, it will be a good source of vitamin D for us. And the oyster mushroom powder, this is oyster mushroom powder, can be added to any food, any recipe, even for making your golden latte. I think you may be aware about latte because it is very famous coffee house people are preferring out of India mainly. So these are the value addition of mushrooms. What all products can be made from mushroom powder and mushroom? This is mushroom cookies you can make, mushroom toast you can make, mushroom bread, cupcakes, mushroom buddy nuggets, pickles you can make, papad, candies. These all are the value added products. We have developed standardized most of them, and this is the product loved by most of the people. We have developed this is some more, and it is made up of mushroom powder and moringa leaves. So this tastes just like Maggi masala. So once you keep a pinch of it on your tongue, you will say that this is Maggi masala, but actually it is made up of mushroom powder and moringa, and the seasonings which we use, herbs and spices which we use in our kitchen, no color, no preservative. All the colors are made from curcumin, that is haldi, and your red chili powder. These are the natural colors in that, and it is also containing, it is a very scientifically designed product. Although it tastes like Maggi masala, but whatever ingredients are present are for some purpose. Like vitamin D, we know in mushrooms is present, but once we don't have fat in our diet, zero fat, then it will not be absorbed, because it is a fat soluble vitamin. Vitamin A, D, E, K are fat soluble vitamins. So vitamin D absorption is enhanced by adding sesame oil and mustard oil in this product. So this kind of innovations can be done by you also. I'm just showing this to have an idea. So it can be used in the noodles, your oats, and anything. These are dry mushrooms. These can be directly added to any curry also, in the soups also, and also you can soak it for some time, just like the nuggets, nutri nuggets of soya. We prepare the curry. You can make the curry. Even Sanjeev Kapoor, if you will type on Google, you will find a recipe of biryani by Sanjeev Kapoor on dry oyster mushrooms. So this is another innovation we did. This is the biodegradable packaging trays, and some more things are done from us. These are the materials developed from by us, by Betty. We have got many awards for all these kind of works. I will show you. This is all made from waste generated after mushroom cultivation. So this is being used for pack. Although these are like handmade products currently, and we have got a grant for develop the machines for this, and we are under progress for this, but we have good orders for this kind of products. This keychains, this is the raw form, and this is the customized form asked by our corporate customers. These all products are developed. My daughter also works on this. So these are the awards. Last year we got, we were the winner of BNest Smart City Bhopal, and this year we got first prize in IIM. We were the winner of I5 Summit because of the innovations and blessings, and these are the achievements we have got. And recently, Rajya Sabha TV has already covered a story, and recently this Aaj Tak team also, as ma'am has already announced, that Aaj Tak you will be very shortly, they will be releasing the videos and all, so I will share. So this is all about Betty, and success is not final, failure is not fatal, it is the end, it is the courage to continue that counts. Thank you very much for providing me this wonderful opportunity and share and learn from you all. Thank you.
Is there any questions? Are there any questions from anybody? Okay. So thank you, ma'am. Thank you for such a knowledgeable and vast lecture. I can't even express, and I don't even have any words to tell you how knowledgeable it was. And there were so many things in your lecture that even we didn't know till now, that how these mushrooms, not only eating, but it will also, eating the mushrooms will also help to preserve and save the environment at the same time. So I think whenever we will eat mushroom, we can proudly say that we are saving our environment. Yes. So thank you so much, ma'am. So,
thank you for listening so patiently, and I'm happy that I was able to create little bit awareness and make you people, many people understand about not only eating, and also for encouraging them for growing mushrooms.
Sure. Thank you, ma'am. And with the end of both the lectures of today's session, may I now request Professor Vinodhar sir, Coordinator, Sri Vaishnav Institute of Agriculture, Indore, to propose words of thanks.
And resource persons of the session, Dr. Ashok Jadhav and Dr. Pooja Dubey Pandey. The lectures were quite insightful, very knowledgeable, and I hope it has benefited the faculty. It has a scope, academic scope. You can go for researches. It has a scope in industry. The nutraceutical, the nutritive, and the cosmetic values that it carries with, that you depicted in your lectures, the method of growing of button mushrooms and oyster mushrooms, and the value addition which we can do with the mushrooms. So what needs to be done is to create awareness amongst the cultivators. As I see in Indore, the mushrooms are not that popular. You don't see mushrooms in market, local markets. It's only available in the supermarkets. So we need to educate our cultivators about this. And the consumers also. As you said, they have the belief that it is non-vegetarian. Whereas the fact is that it is vegetarian. So we need to spread awareness, and this institute has established an extension wing which goes to the villages and the panchayats to spread the awareness about many other things and about the mushrooms also. At our institute, we have been growing oyster mushrooms of different species. Butter mushrooms we tried, but it is most temperature sensitive. So you need to grow these in controlled conditions. But we have got little success in butter mushroom, but we are very good at growing of oyster mushrooms. So thankful, I am very thankful to both of you for this very knowledgeable lectures. And I believe and quite hopeful that my faculty at this institute has been benefited well. It is a scope, good scope for research for our department of pathology. The mycologists have a scope. The biotechnologists have a scope. We have department of biotechnology also. So they have a scope. Academically, they have a scope. And the students, they who pass out from this institute, they can start up their own ventures. They have a scope. So thank you very much, both of you. It has been nice listening to you. Thank you very much.
Thank you, sir. Thank you for the nice
Thank you.
words.
Thank you, sir, and thank you all for your presence and patience. And with the permission of Dean, Faculty of Agriculture, I conclude today's session. Thank you and have a nice day.
Thank you very much. Thanks again.